Thank you, our esteemed guests. Now we would officially commence our conference with the opening of the abstract book of our conference. I would like to request our honored guest to do the honor. Sirs, please.
With the opening of the abstract books, we have commenced our conference officially. A huge round of applause. Again, a huge round of claps, everybody.
Thank you,
Thank
sir.
you. Thank you, sir. Now our students will set the mood of the atmosphere of our conference with their musical performance. I'm requesting our students to please come in front of the stage and enthrall us with their musical capabilities.
Hello. জাগরণে যাইবি ভাবরি আখি হতে ঘুম নীল হরি কে নীল হরি মরি মরি জাগরণে যাইবি ভাবরি আখি হতে ঘুম নীল হরি কে নীল হরি মরি মরি জাগরণে যাইবি ভাবরি the person for whom I roam alone my eyes crave for him yet he is nowhere the person for whom I roam alone my eyes crave for him yet he is nowhere his melody is his melody his melody plays in the depth of my heart oh my entire night passes thinking about him জাগরণে যাইবি ভাবরি বাণী নাহি তবু কানে কানে কি যে শুনি তাহাকে বাজানে বাণী নাহি তবু কানে কানে কি যে শুনি তাহাকে বাজানে এই হিয়া ভরা বিহনদে অশ্রু আহে মোর নয়নতে এই হিয়া ভরা বিহনদে অশ্রু আহে মোর নয়নতে ছায়া দোলে তারি ছায়া দোলে দিবানিশি ধরি মরি মরি জাগরণে যাইবি ভাবরি আখি হতে ঘুম নীল হরি কে নীল হরি মরি মরি জাগরণে যাইবি ভাবরি I wonder who had taken away sleep from my eyes yes sleep from my eyes মোড়ো মোড়ো জাগরণে যাইবি ভাবরি
A wonderful performance by our students, A big round of applause for them. Now it's time to flow with our conference schedule. I am requesting Professor Nirmal Kumar Sarkar, Emeritus Professor, Department of Biological Science, Midnapore City College, to give the keynote speech and welcome our esteemed delegates. I am requesting sir to please come to the stage.
মাইক যেন ঠিকঠাক থাকে, বারে বারে ডিস্টার্ব করবে না। চালু আছে? চালু করো। চালু আছে? Honorable Founder Chairman of this college, Sri Prabhas Ghosh, Honorable Founder Director of this college, Dr. Pradeep Ghosh, respected Principal, Dr. Sudipta Chakraborty, respected Vice Principal, Dr. Kuntal Ghosh, distinguished resource persons and guests from different institutes, learned colleagues, and organizing committee members, dear staff members, researchers, and student participants. It's my proud privilege to make a prelude in this inaugural session of the 5th Annual National Scientific Conference of Midnapore City College. At the outset, I would like to express sincere thanks and gratitude on behalf of all members of this college to Sri Prabhas Ghosh, the founder chairman of this college, and to Dr. Pradeep Ghosh, the founder director of this college, for their kind participation in this inaugural session. Side by side, I extend hearty welcome to all the resource persons, guests, teachers, researchers, and students participating in this conference from this college and other institutes. Now I would like to brief everyone in the hall, especially the students, about our college. Our college happens to be the first self-financing undergraduate cum postgraduate college in South Bengal. It is affiliated with both Vidyasagar University and West Bengal University of Health Sciences. And it is recognized by the Higher Education Department as well as the Directorate of Medical Education of Government of West Bengal, and also by the University Grants Commission as well as the Department of Scientific and Industrial Research, Government of India. The foundation stone of this college was laid on 6th February 2017, and the college started functioning in a single two-storied building on 10th August of 2017 with just 134 postgraduate and 20 undergraduate students. That is 154 students in total. But there have been dramatic changes, both quantity-wise and quality-wise, in the scenario within the last 7 years. All these changes are primarily due to the untiring efforts, enviable tenacity, and farsightedness of Dr. Pradeep Ghosh, who is not only the director, but also synonymous with the heart, lung, or the brain of this college. I take the opportunity of this inaugural session to gladly mention here that just 1 week ago, Dr. Pradeep Ghosh has been awarded a prestigious leadership award by the Chirantan Rasayan Sanstha in collaboration with the Royal Society of Chemistry of England in a meeting at Christ University of Bangalore. As compared to a single two-storied building in 2017, the college now spans across 3 multi-storied buildings accompanied by a few small annexures. Again, as compared to only 154 students in 2017, the student strength reached a total of 6,186 in November 2023, which included 3,700 undergraduate and 2,486 postgraduate students. At present, the college teaches as many as 19 postgraduate and 17 undergraduate courses under Vidyasagar University. And additionally, the college teaches 4 postgraduate and 2 undergraduate courses under West Bengal University of Health Sciences. Moreover, this is the only college in the 2 Midnapore districts which teaches 10 job-oriented courses on agriculture, fishery science, hospital administration, medical laboratory technology, medical microbiology, nutrition and dietetics, food science and nutrition, plant pathology, computer application, etc. It's my pleasure to mention here that as of 31st January 2024, as many as 1,907 of the present students are recipients of Swami Vivekananda Merit Cum Means Scholarship from Government of West Bengal. 818 girl students are getting Kanna Shri Part 3 scholarship from Government of West Bengal. 122 are getting Oiko Shri scholarship from Government of West Bengal. 525 are getting national scholarship from Government of India. 62 are enjoying Oasis scholarship from Government of India, while another 110 students have received various other scholarships. Overall, 3,544 or 57.3% of the total 6,186 students are enjoying one or another kind of scholarship. It is also worth mentioning here that in the last 3 years, 14 of our postgraduate students qualified the NET, 7 qualified the SET, 10 cracked the GATE, and another 3 cracked the GEST. We are also happy because some of the alumni of this college could ensure their placement as researchers in reputed institutes of South Korea and Germany, in addition to IIT Mumbai, IIT Delhi, and IISc Bangalore. At present, we have more than 25 well-equipped laboratories containing various research instruments. Since December 2021, Vidyasagar University has sanctioned PhD research center status to our college. Till now, 34 research scholars have already got their registration from Vidyasagar University for their PhD pursuit under the supervision of different learned teachers of this college. They get scholarship from different funding agencies. Moreover, 10 prospective researchers are now under training process and receiving monthly Moraine Research Fellowship from the college itself. indeed happy to add here that 6 talented teachers of our college have been utilizing research grants over 4.69 crores of rupees from different funding agencies for implementation of their research projects. Chronologically speaking about them, Dr. Shravani Pradhan, the coordinator of nutrition-related sciences and also the organizing secretary of this conference, is an ardent researcher on development of nutraceuticals from local marine fish sources for prevention of cancer and obesity. Dr. Pradhan was the first teacher in this college to receive an ICMR grant of rupees 24 lakhs, and later she received another ICMR grant amounting rupees 44 lakhs. Dr. Pradhan was honored with the prestigious Hope Fellow Award by Japan Society for Promotion of Science during her DST-sponsored visit to Japan as a young scientist in 2016. Dr. Kuntal Ghosh, former postdoctoral fellow in Department of Food Science and Technology of Chonbuk National University of South Korea, and now vice principal of this college, is a brilliant researcher in various fields of medical microbiology with emphasis on probiotics technology. He also works on genetics and molecular biology of bacteriophages. Dr. Ghosh initially utilized a DST SERB grant of rupees 35 lakhs and is now utilizing an ICMR grant amounting rupees 41 lakhs. Dr. Ghosh also bagged the Best Paper Presentation Awards twice in South Korea and once in Sikkim. Dr. Suchishmita Roy, a diligent teacher of nutrition-related sciences, is an ardent researcher in the field of detection and management of renal disorders. Dr. Roy also had a brief experience of research pursuit in the University of Plymouth in England. Dr. Roy has received an ICMR fund amounting rupees 44 lakhs for working on early detection and phytotherapy of renal disorders induced in murine models. Dr. Shantanu Kaur Mahapatra, Head Department of Paramedical and Allied Health Sciences, is a brilliant researcher on immunity to infectious diseases. He owns an international patent for an anti-leishmanial compound. Dr. Kaur Mahapatra, as the principal investigator, along with Dr. Shravani Pradhan as the co-PI, have received a massive grant of rupees 88 lakhs from ICMR for detection and cure of tuberculosis among the tribal communities of the entire Midnapore belt. They have also been sanctioned another ICMR grant of rupees 50 lakhs for working on liposome-mediated delivery of anti-leishmanial drugs. Dr. Kaur Mahapatra has also been sanctioned a ACRB core grant of rupees 48 lakhs for isolating anti-leishmanial agents from sugarcane waste. Dr. Sudipta Chakraborty, former postdoctoral fellow in Department of Neurological Science of Rush University Medical Center of Chicago and in Masonic Cancer Center of University of Minnesota, Twin Cities of the United States of America, and now principal of this college, is an ardent and experienced researcher in various fields of therapeutic neuroscience and molecular oncology. Dr. Chakraborty has received an ICMR grant of rupees 47 lakhs for conducting his investigations on neuroprotection in neurodegenerative diseases. Besides, Dr. Shubhendu Kumar Manna an energetic assistant professor of physics, has received a DST SERB grant of rupees 46 lakhs for conducting his investigations on transport property of some specialized magnetic systems. Now turning all your attention to this ongoing conference. Director of this college, Dr. Pradeep Ghosh, all along dreams of escalating the standard of this college to the level of a center of excellence. In parity with this motto, Dr. Ghosh started to organize a national level scientific conference every year, starting from 2018. The recent COVID pandemic broke the chain in 2021 and 2022, but the rhythm was restored in the last year. This is how we are now attending this 5th annual national level scientific conference of this college, organized with grants received from DST, SERB, DBT, and CSIR of Government of India. We deeply acknowledge to all the 3 funding agencies. The theme of the conference, this year's conference, is functional food as dietary intervention for chronic diseases. The term functional food was coined in 1984 in Japan by an 82-member national project team. Functional food means foods that have been processed or modified through enrichment or fortification or application of agricultural, enzymatic, chemical, or any other technological means, so that such foods can provide disease-preventing or health-promoting benefits in addition to their nutritive values. Nutrition scientists across the globe have reached the conclusion that the use of different functional foods can effectively prevent various chronic diseases, prevent side effects of pharmaceuticals, promote health, and reduce health care costs. In this conference, valuable deliberations will be made by a galaxy of invited scientists on the development, utilization, benefits, challenges, and prospect of different functional foods. In addition to the invited lectures, 20 research papers will be presented by research scholars and teachers of this college and other institutes. Besides, around 40 posters will be presented by the enthusiastic students of different institutes. It is our proud privilege to have amongst us Dr. Anil Kumar Anal, Professor and Chair of Food Innovation, Nutrition and Health in the Department of Food, Agriculture and Bioresources in the Asian Institute of Technology of Thailand. Professor Anol belongs to the top 1% scientists in the field of food science across the globe. His expertise lies in fields like food and nutrition security, innovative food processing, and green technology-based valorization of agro-residues. He has an enviable biodata, including authorship of over 200 research papers, 60+ book chapters, 10 edited books, and 7 patents, along with research experiences in Otago University and Massey University of New Zealand. He has been acting as research advisor cum consultant in a number of Asian as well as European countries. We extend hearty welcome to Dr. Anal, who has kindly taken the trouble of traveling over 3,000 kilometers from Thailand to Midnapore to enrich us with his valuable deliberations. We are lucky to have amongst us Dr. Debaprasad Chattopadhyay, former Managing Director of ICMR Virus Unit, Kolkata, Director of ICMR Regional Center at Belagaum, Karnataka, and Founder Director of ICMR National Institute of Traditional Medicine, Belagavi, Karnataka. His expertise lies in ethnomedicine, virology, microbiology, and molecular medicine. He has an outstanding biodata, including publication of 135 research papers, 5 edited books, 26 book chapters, and 7 patents, receipt of overseas fellowship for postdoctoral work in London Hospital Medical College and Statens Serum Institute of Denmark, and also receipt of several prestigious awards. We welcome Professor Chattopadhyay most cordially to this conference and look forward to his valuable deliberations. We shall be lucky to have amongst us Dr. Madhumita Barua, Professor of Agricultural Biotechnology in Assam Agricultural University. Her expertise lies in fields like plant microbe interaction, food microbiology, genetic engineering, and metagenomics. She has a wonderful biodata, including over 160 research publications, receipt of Fulbright scholarship for having pre-doctoral training in Texas A&M University of America, and Commonwealth scholarship pursuing PhD work in University of Exeter in England, and also the receipt of several prestigious awards. We all extend warm welcome to Dr. Barua and eagerly wait for listening to her illuminating deliberations. It is our proud privilege to have amongst us Dr. Keshav Chandra Mandal, Professor of Microbiology and Biotechnology in Vidyasagar University. He is an expert in microbial enzymology, probiotics technology, ecology of gut microbes, and exploration of natural antimicrobial agents. Dr. Mandal has an outstanding biodata with publication of over 160 research papers, 8 reviews, 23 book chapters, and a textbook. Professor Mandal is the owner of Chancellor's Gold Medal at university level and subsequently Indo-Hungarian Fellowship, Tempus Public Foundation Fellowship, and a few other awards and fellowships. It's also worth mentioning here that both Kuntal Sir, that is Dr. Kuntal Ghosh, and Shubhadeep Sir of Microbiology Department of this college carried out their PhD pursuit under the supervision of Dr. Mandal only. We fervently welcome Dr. Mandal and eagerly wait for his informative deliberations. Oh, onek din bajben. Long live the king. It is indeed our pleasure to have amongst us Dr. Dept Bhattacharya, Scientist E-cum Deputy Director of Regional Medical Research Center under ICMR at Bhubaneswar. His expertise lies in microbiology, epidemiology of infectious diseases, antimicrobial resistance, and public health. He has nearly 100 research publications to his credit. He is the owner of ICMR Centenary Gold Medal for his outstanding research contributions in the field of shigellosis in Andaman and Nicobar Islands. We cordially welcome Dr. Bhattacharya and eagerly wait for his valuable deliberations. It will be our pleasure. Now it is time to welcome Professor Dilip Kumar Nandi, an eminent professor cum scientist from Midnapore itself. Professor Nandi, the son of the soil, headed the physiology department of Raja N.L. Khan College of Midnapore for years and is now attached with this college as Professor of Paramedical and Allied Health Sciences. Professor Nandi is a veteran researcher on development of nutraceuticals, isolation of phytomedicines, and isolation of probiotics with therapeutic potential. A glaring example of success of the teaching career of Professor Nandi is that both Dr. Shravani Pradhan and Dr. Suchishmita Roy of our college had earned their PhD degree under the direct supervision of Professor Nandi only. You are most cordially welcome, sir, in this conference. Finally, I take this opportunity once again to welcome all the resource persons, guests, as well as teachers, researchers, and students participating in this conference from this college and other institutes. I believe this conference will ensure a flow of knowledge as well as blessings from the top order to the bottom order, or from the experts to the novice. Till then, I wish you all a nice time in this college. Thank you very much.
Thank you very much, sir, for your valuable and informative speech. Now, I would like to request our madams to felicitate Dr. Keshav Chandra Mandal, Professor, Department of Microbiology, Vidyasagar University, on come onto the stage. Now, I request all our renowned and honorable guests to take their designated seat as our technical session will going to start. Sir, please take your seat. Now it's the time for our technical session 1, and it will be chaired by Dr. Dilip Kumar Nandi, Professor, Department of Paramedical and Allied Health Science, Midnapore City College, and former Associate Professor and Head and Coordinator, Department of Human Physiology, Food Science and Nutrition, Microbiology, and BMLT Raja Narendra Lal Khan Women's College, and also former executive member and coordinator of Netaji Subhash Open University. He has been serving as guest and visiting faculty member in the postgraduate studies of physiology, nutrition and dietetics, biomedical laboratory science and management, biochemistry, biotechnology and zoology of Vidyasagar University, as well as different institutions since 2002. Presently, there are 22 PhD students under his guidance, and 16 have been awarded with PhD degree from University of Calcutta, Jadavpur University and Vidyasagar University. He is also home supervisors of Newton Bhava PhD placement program under British Council of Plymouth University and Leeds University. His area of research is alternative management of kidney disease and health promotion through functional foods, nutraceutical, herbal, nanotechnology, yoga and exercise. He has published more than 80 scientific research articles in peer-reviewed national and international journals, and also several book chapters in UNESCO, Elsevier, Taylor and Francis publication groups. He has been completed as PI of different major, minor inspire research projects sanctioned by UGC, DST, DRDO, DIPAS, and also organize several national and international conference, seminar, and workshop sponsored by UGC, DST, DRDO, SERB, DBT, and NAS. Dr. Nandi is a former Vice President of Physiology Society of India and South Asia Association of Physiologists. He was awarded BB Sarkar Memorial Oration and Founder Fellow of PSI, and also awarded Social Service Certificate in the cause of humanity through Medicare Rescue and Relief Work organized by St. John Ambulance Association, West Bengal State Center. He had served as Honorary District Secretary for Bharat Jano Gyan Vigyan Monch, organized by National Council for Science and Technology Communication under DST Government of India in the year 1922. Sir, please, I would like to request you for chairing the Department of
Good morning, everyone. Very nice morning today, and very nice speakers and the resource persons are present. At first, I convey my deep sense of gratitude all of you for your presence annual conference. I do not like in the national. It is international. Also the conference make it in this way, and very importantly, the topics is very relevant to our society nowadays. Very relevant. It is undoubtedly, as because in inauguration session already mentioned by our honorable respected officer Nirmal Kumar Jana, he is a very veteran teachers, and also he as a teachers of the teachers in retires from the Presidency College. I am very much proud for him. Actually, he is a teacher in my along with my teachers in a very dynamic teachers. In particularly, he lost in last December 2023. Officer Devojyoti Das, you know all. Okay. Now, these sessions actually theme of this session, according to the organizer of this conference, is is in this technical session mentioned by the organizations according to the thematic discussions within this range, in particularly the modern weapon using the dietary component to combat chronic disease. There are a lot of diseases, and this in this technical session, very informative, innovative scientists and the teachers are present here or deliver their speech or discussing their content. And first is Professor Devi Prasad Chattopadhyay. Already you know the Professor Devi Prasad Chattopadhyay. After that, Professor Keshav Chandra. I am very much about for the Keshav Chandra Mandal and Professor Chattopadhyay. But I mentions or introducing before you for Professor Dev Prasad Chattopadhyay, and after that, and Keshav Chandra Mandal. That will be the better. And Professor Dev Prasad Chattopadhyay, a biomedical scientist, really reputed scientist. Now, according to your credibility, he scope up in Google Scholar score citation in more than near about 6,000. Not a matter of joke. His dedications and also the devotions in the promotions of the disease promotion, particularly in the disease promotion in different way. And as a scientist, I am very much proud about Chattopadhyay, the scientist, the second positions in the 10 scientists of the ICMR, along with the 10 out of 10 in the ICMR scientists. His position second rank, and also among top 20% global scientists rank. Is not a matter of jokes. It is a very really sad. We are very much proud to you our presence here. And he has completed his postgraduate degree in the botany in the microbiology from the University of Bardhaman. Okay, sir. And also the completed PhD from the Jadavpur University, the Department of the Pharmacy, the year 1989. And apart from this, then moved to the Royal Hospital, the London Hospital of the Medical College, London, for postdoctoral training on the clinical trial of the phosphomycin, and also the role of non-antibiotics in bacteria at State and Serum Institute in Copenhagen in the durations of the period is 1919 and 1993. Then on on returning back, Dr. Chattopadhyay joined the CSIR, Indian Institute of Chemical Biology, as CSIR Senior Research Associate. He started his scientific journey at the ICMR Regional Medical Research Center, Port Blair, in December 1993, and shifted to the ICMR Virus unit in Kolkata in 1997. Dr. Chattopadhyay was selected as the first director of RMRC, the Belgaum, in the year 2016, which was upgraded to the ICMR National Institute of Traditional Medicine in the year 2017, where he served as the founder director, even also the scientific G, until the September 2021. Presently, he is associated with the director and School of Life Sciences, Swami Vivekananda University, Barakpur, Kolkata. After serving at the School of Health Sciences in NSHM Knowledge Campus in Kolkata and Durgapur as research advisor. Dr. Chattopadhyay doctoral work established the antimicrobial potential drug of the dialysin in vitro, even also the in vivo process in the year 1988, and that the revert the drug resistance by the altering of the membrane permeability of the bacteria. His postdoctoral training was on the clinical trial of the phosphomycin in uncomplicated UTI involving 84 GS, covering the England, even also the Ireland, and also the Scotland, under Professor J. Williams. Then in the year 1994, early 1994, has initiated the validations of the healthcare practices and also the use of the tribal traditional medicine of the Bay Island, using the term of the ethno-medicine. Dr. Chattopadhyay demonstrated the bioactivity of 17 traditional formulations with standardization, the fractionization, isolation of different phytomolecules to establish the purity, safety, even also the potentiality, and also the preclinical efficacy in animal models. A series of publications already mentions our Professor Sarkar. Using the ethno-medicinal approach, Dr. Chattopadhyay has identified the molecules having the significant antimicrobial, antiviral, even also anti-inflammatory, and also modulatory, contraceptive, and wound healing, iron chelation, and abilities, with different publication contest. He is supervised till now in 20 PhD scholar out of 20 already awarded. A very few, few introductions of the Professor Chattopadhyay, and so many awarded and honored he has credited, and also handling so many research, handling so many projects, major or the minor project, funded by IUS, DST, SER, SERB, DST Women Scientists, very good, and also the DST Task Force, NIF, DST, ICMR, and DBT. Thank you, sir. And very innovative and interactive topics.
Very good morning to all of you. First of all, I convey my sincerest gratitude and deep thanks from the core of my heart to the authority of this. Professor Nandi, thank you so much for your elaborate biodata, presentation of my biodata. And particularly, I know some of the faculties of this institute, and you see, few of my students who did PhD under me, and mostly all the 20 PhDs, those who have done PhD under me, all are well established. And one of the student was from Paschim, Medinipur. He is now in National Institute of Health. And I am very much proud of that person, because it's not very easy to get a chance in the as a faculty of that particular institute, National Institute of Health, America. Similarly, all my PhD students are well established, but the thing is that out of my nearly 30 years career, I have learned a lot of things, and when I started validating the traditional medicine of the tribes of India, particularly the oldest tribes of India in Andaman Nicobar Island, way back to 1994, you see, those traditional healthcare practices have been documented and demonstrated by our group to the world. Out of 8 patents, I have 2 US patents, and those patents are accepted without any modification. That is another beauty. I have few publications without modification. The publications or paper was published, like contraception, like PLOS pathogen. So these are the Journal of Biological Chemistry, because this actually indicate the commendability of the work and the in-depth knowledge of this. But today's topic is functional food, nature's weapon to counter chronic inflammation leading to chronic diseases. You see, I have more than 24 publications on inflammation related aspect in different disease condition. Out of that, I found that chronic inflammation is the main reason of all the diseases. You see, when there is a disease, there should be inflammation. And that's why in any disease model, inflammation should be tested. So inflammation is the root cause of any of the devastating diseases. You can classify the disease in 2 ways. One is the infectious disease, other is lifestyle disease. বাংলাতে যদি বলেন তাহলে রোগকে দুভাগে ভাগ করতে পারেন, একটা জীবাণু ঘটিত আর একটা জীবনজনিত। মাইন্ড দ্য টার্ম জীবনজনিত। সো উই ক্যানট এবল টু এলিমিনেট দ্যাট জনিত শব্দটা, কেননা মানে জন্মার সাথে সাথে বড় হবার সাথে সাথে আমাদের এই রোগগুলো হবে, লাইফস্টাইল ডিসঅর্ডার, বিকজ you cannot maintain the whole homeostasis of your body. When once there is a chance of dismantling the homeostasis or changing the homeostasis, then inflammation starts. But the question is that if you allow the inflammation to go for a chronicity or chronic condition, then only disease comes. So the next slide. Now chronic diseases, accounting for about 70 to 80% global death. These are characterized by low grade long-term inflammation, ultimately altered metabolism or organ damage. Actually, I am not telling anything new here. I am telling the research finding and many of the things you know. So I am trying to deliver the talk in a way so that the students can understand. Okay, there are huge number of slides. I may escape some of the slides because our work is also there. Okay. Long-term silent inflammation in living system may lead to incurable chronic condition, and thus chronic low-grade inflammation is the major cause of chronic diseases. It is well established, not new thing. Now inflammations are the immune system response to any harmful stimuli. বাড়ি গেলে আমার কান মলে দিত, বলতো তোকে দিয়ে কিছু হবে না, এটা বাংলাতে বলতে সুবিধে বলে বলছি। 2-3 বার এরকম হওয়ার পর আমি খোঁজ রাখতাম কখন ওই মামা আমার বাড়ি আসে, যেই আসতো আমি পালাতাম খবর পেলে। That is called resistance. এই রেজিস্ট্যান্সটা ন্যাচারাল, কেমন আমাদের ইমিউন সিস্টেমেরও এই এবিলিটি আছে। So long term chronic inflammation leads to global disease, global death, 70 to 80% global death. An inflammatory response begins with the production of release of certain chemicals by the infected or injured or diseased tissue. They are characterized by the vasodilation, blood vessel dilation hoy, increased capillary permeability, swelling, clotting of intestinal fluid, and migration of monocytes and granulocytes. I will explain these things step by step so that you can understand, because immunity is just like any country's defense. But our immunity is much more strong than the American defense system. Okay. I will explain one by one because I learned immunology from a very talented scientist, Dr. Bhaskar Saha, who is actually the Bhatnagar awardee scientist and one of the top immunologists of the country. I have a close association and I learned immunology from him, and he learned virology from me because I am one of the senior most virologists of the country. So these are the etiology of inflammation, physical factor and infections. Now functional food, already you are working. Functional food means the part of the food that is functional in the body. It may be exemplified or terminate, or terminology is different. Maybe phytonutrients, functional food, phytomolecules, anything you can say. Which is functional in your body. You see, which is functional, they also can able to do harm. It's not that all functional food are good. When there is work, if something work in your body, something against the work is there also. Just like Newtonian principle, law of action and law of inaction. So modern science is looking into the healing power of dietary ingredients. Why? Because nobody is used actually when I was the demonstrator of London Hospital Medical College, I teach MD microbiology. You see, in London microbiology MD is done only by doctors. MBB microbiology MSc and MD both. There is no other students cannot able to enter there. So as a postdoctoral fellow, I had the duty to teach them animal modeling and taken class of medical microbiology. So I learned a lot of things from there. You see, inflammation regulating food. Nowadays we have to include a lots of, WHO is also asking about lot of fruit, 9 servings of fruits and vegetables on your diet, but most of the people do not take that. And secondly, all the fruits and vegetables are adulterated in such a way, the pesticides, herbicides, a lot of things. And in 2016-17, I used to write article in all of the national dailies, starting from Anandabazar Potrika to Telegraph to Deccan Herald. Everywhere my article was published. Why do you need supplementation to overcome the conditions so that you cannot get the inflammatory diseases easily. So supplementation, and some people used to tell about supplementation also. So I'm not telling about supplementation here. The functional component of specific food that actually interact with the oxidative and inflammatory pathway, that is important. We have oxidative and inflammatory pathway generating ROS and nitrogen species also. Here I will highlight along with the clinical trial and selected dietary ingredients that alter inflammatory and metabolic pathways or mediators. So this is the objective. I have already told a number of functional food groups and critical enzymes and their bioactivities altogether, how they protect synergistically or additively, because my target is to show the mechanism of action of some of the dietary ingredients, so-called nutraceuticals, published in JBC and all other very, very big journal here. Okay. So functional food, you already know, the functional food are food that prevent disease. In true sense, it was Harvard Health Letter, April 2005. And building block of functional food are phytochemicals or phytonutrients, the natural bioactive substances that prevent diseases by interacting with the body's innate healing power to maintain vibrant health. Actually, the definitions are very closely related. These definitions are very closely related. So functional foods are the dietary compounds or components containing bioactive phytonutrients, dietary fiber, nutraceutical, probiotic, those who have health promoting or healing power. Okay? And at safer concentration, they provide required amount of essential nutrient to the body and health for healthy living and prevent oxidoreductive diseases leading to inflammation. Several such food demonstrated anti-inflammatory and antioxidative properties in diverse cell lines and animal models, but still it is inconclusive in human, not only for trial, but also a trial may give you something, but whether you isolate the component from the body, can you see how much amount of the things absorbed in the body, how they are assimilated and how they are breaking down within the body. This is not very easy. PKPD and everything determination is not very easy in the living system. If we do not get, rather body cannot grow and heal as functional phytochemical or for food provide raw material for healing and macromolecule provide fuel to perform the optimal level. Without sufficient functional food, body can go for starvation, molecular level starvation, not apparent. Okay, toxicity, low level toxicity, stress, another way of generating ROS and nitrogen species, and degenerative diseases. This is also taken from our book. Now what is inflammation? So the word inflammation, I will show what inflammation, normal inflammation and chronic inflammation. Actually inflammation is classified into two forms. One is acute inflammation, other is chronic inflammation. See how they will differ. Body's natural protective response to the invader, either it is pathogen or toxin or any accident, anything. Human immune system cannot discriminate between pathogenic invader or internal things or trauma. They cannot able to discriminate, but they understand self and non-self, and that is the problem. Specific surface protein in our cells, pattern recognition receptor, can detect invader or damage, while immune system respond to both types of attack in the same way. Once realize the threat, immune system starts to move to remove the source and begin healing process. So inflammation is destructive as well as a constructive mechanism. If you go to the definition now, you will find that inflammation is constructive as well as a destructive process. Okay. Immediately blood vessel dilate to increase blood flow, making injury or infection area warm and red. If you got an accident, you will feel all these things. Now capillaries became permeable, white blood cells move to the injured area and cause swelling. Isolate the invader of damaged cells, actually clear clear away the dead cells and damaging substances and begin repair process. This is the healing process starts in the body. And inflammation is actually good for the body because it is the immune response. When body detects any harmful substance, so there is an inflammation means body is in defense mode. Body is in defense mode. Localized at injury site, localized infection or systemic infection. When inflammation is acute or lasting for few days, then body can manage it or even drug can manage it. But when it continue for a long time, there is no drug. So chronic inflammation has no drug. That's why that is very important. And all the diseases are actually the byproduct of chronic low grade undetected inflammation. So these are the 7 factors that cause inflammation, and you see redness, swelling, pain and heat. Okay. Now chronic inflammation, if you consider, develop a part of sequel of cellular events following acute inflammation. I have already told, and chronic inflammatory diseases are most significant cause of death in the world. You see, cancer, diabetes, pancreatitis, cardiovascular disease, IBD. What are the diseases of today's world are due to chronic inflammation. Okay. And persistent immune activation results in chronic inflammation. Persistent activation. কেউ যদি আপনাকে ক্রমাগত উত্তেজিত করে তাহলে আপনি কি করবেন? দেব এটাকে এক থাপ্পড়, তাই না? এটাও তাই, ক্রমাগত। তাহলে সোর্সটাকে রেকগনাইজ করতে হবে। উই হ্যাভ টু আইডেন্টিফাই দ্য সোর্স টু এলিমিনেট ইট। I will give you some clue how to manage yourself to prevent from such type of destructive chronic inflammation for long time. Okay. So these are the acute inflammation and chronic inflammation, the differences between these. Several biological factors or molecular factors are there. It will be time taking, but accumulation of polymorphonuclear neutrophil that convert into macrophage, the formation of edema starts. And damage associated molecular pattern and all other big names are there because these are biological molecules. Only you think your immunity actually responding in such a fashion because it's a very peculiar and very in-depth network of molecules. And chronic inflammation is actually release a number of cytokines and chemokines in the body. Those are and their internal signaling between crosstalk and self-talk actually leads the situation. Active and chronic, in condition wise, the causative agent is pathogen or persistent or irritant or damage, and persistent is continuous rather. Then major cells, basophil, eosinophil, monocyte, your blood cell, WBC actually. Then immediate and delayed, duration few days, then and that is up to months or years, and outcome is resolved. It can be resolved, but chronic inflammation cannot be resolved by the body because it is a chronic sequence. And parameters are triggered by different parameters, then duration, then magnitude, outcome, age related. Whether it is age related, no, it is not age related. And biomarkers, different biomarkers. If you want to study inflammation, you have to study certain biomarkers. There are several biomarkers, but not only CRP and ESR. Erythrocyte sedimentation rate and C-reactive protein are the 2 good biomarkers, but those are not the sole biomarker. In many conditions, these are modulated or be high. So other biomarkers is also there, interleukin, TNF alpha, and in case of systemic inflammation, silent, no canonical standard biomarker is detected till date. But these biomarkers may be altered in chronic inflammation also. So you can, if you do not understand from this, from sign you can understand. Flushed skin at the site of injury, fear of tenderness, swelling, and it heals. This is the acute inflammation. In acute inflammation, there are anti-inflammatory drugs, but in chronic inflammation, nothing is there. It will not work. Okay. And chronic inflammation may be abdominal pain, maybe chest pain, maybe rash, maybe other things, fatigue, fever, duration. If you go to doctor will tell you to test a battery of events, and you will test. From test what you will get? maybe intermediate, because you see any parameter there is a lower value and upper value, and those value is not determined by the Indian population. For the Indian population, it is either Caucasian or the company, those who created the kit or manufactured the kit, they give you the cut point, cut off value. So that does not mean yours are the same. So what happened? Doctor will tell nothing is happened. Then if we go to a doctor and always expect certain drug, so doctor will prescribe some medicine, anti-inflammatory or vitamin, minerals, take rest, go for good sleep. After 10-15 days when drug is covered, you will go to the same circle, same cycle of events, of same cycle of work, and again the things come, because the root cause of this type of diseases is your dietary pattern, is your chronic inflammation that has not been detected by any medical test. So you will go to the vicious cycle. Okay. So this is the inflammatory response after an injury, you see, wound or skin, then or bacteria, or say histamine release and the phagocytes wanted to kill it, and then goes the several events. Ultimately non-specific defense work and leads to vasodilation and increased vascular permeability, phagocytosis and destroy microbes, and outpouring plasma due to increased vascularity, a fibrin barrier minimize the infection. This is actually in acute infection or acute inflammatory response. But immune response in inflammation against pathogen can be elaborated, and it has a lot of publications, a lot of studies there. If you see, this is the pathogen, and this is the host defense is trying against it. This is tissue damage, so interleukin is released, and a lot of parameters from different cells, different tissues or different organs are going to play together. Several factors play together. Then transition, physical, chemical or biological stimuli go for injury, acute inflammation or vascular damage, then leukocyte recruitment, then mediator release, and the healing process is this, it goes resolution. But if acute inflammation do not resolve, progress to no improvement, then it goes for chronic inflammation. And chronic inflammation means these things happen: angiogenesis, blood vascular formation, cell proliferation, polymorphonuclear leukocyte, fibrosis, and the beginning of certain diseases. But it takes years. On a single day, this type of disease do not form. It takes 15-20 years to develop a disease. And for cancer, I'll tell you very basic things. I do not know whether you know or not. Every human being contains approximately 10 to the power 13 cells. And scientists reported that out of 10 cells, 1 cell is defective or cancerous. So in 10 to the power 13 cells, what happened? 10 to the power 13 minus 10 to the power 8 means 10 to the power 5. At any moment of time, if a person has got 10 to the power 5 cell defective or cancerous, that person will be detected as a cancer patient. But body has a very peculiar mechanism that is called apoptosis or programmed cell death. Each and every day, body destroy or kill nearly 10 to the power 4 cells. The apoptotic mechanism in whose body the apoptosis is good, they may not develop those things. But in many persons, apoptosis may not be very correctly run, because apoptosis depend upon your mental status, your daily life, your food, your thought process, everything. And that is very difficult to control. Okay. Now, what cause inflammation? Autoimmune disorder, exposure to toxin, untreated inflammation, autoinflammatory disorder, repeated episode of acute inflammation, বারবার ধাক্কা লাগছে, বারবার পুড়ে যাচ্ছে, mental stress, lifestyle factor contribute, excess alcohol drink on regular basis, high body mass index in the range of obesity, exercise at maximum. কেউ কেউ আছে না রোগা হবার জন্য দুরন্ত এক্সারসাইজ করছে, কি করেছি দেখো, কি 36 প্যাক থেকে 26 প্যাকে নেমে গেছে, কিন্তু ভিতরে প্যাকাপে কি হয়ে গেছে তখন। They do not understand. Heavy exercise and chronic stress and smoking or exposure to long exposure to smoke irritate your cell, and cell undergoes destructive process and inflammation continue in a low level. So So what is immunity? Now the word comes, it's a very interesting thing. Ability of an organism to resist an infection or toxin or specific antibody or sensitized WBCs. Body's ability to recognize germs and prevent them to cause illness, we all know na. But what is immune system? A systematic approach or system, just like our defense system. Defense minister থেকে শুরু করে অনেক আছে, হ্যাঁ আমরা কত কত কামান থেকে শুরু করে সব কিনছি, বডির জন্য কি কিনছি? বডির তো একটা ডিফেন্স আছে। দেখুন বডির দুটো পার্ট, একটা উপর পার্ট একটা নিচের পার্ট। নিচের পার্টে আমরা খুব ডেকোরেটিভ থাকি, উপর পার্টের ডেকোরেশনটা কি? রেগুলার বই পড়া, রেগুলার ভালো কিছু শোনা। আমরা নাচা কোদাতে ব্যস্ত হয়ে পড়েছি, তাই না? টিভিতে অ্যাপিয়ার করছে, সব অ্যাওয়ার্ড পাচ্ছে, দেখুন দুটো সিনেমা করছে, বড় অ্যাওয়ার্ডই হয়ে যাচ্ছে। কজন সায়েন্টিস্টকে আমরা অ্যাওয়ার্ড দিচ্ছি? সোসাইটি কোন জায়গায় যাচ্ছে দেখতে হবে, কেমন? So those are the things. Immune system help to identify and eliminate the germ that entered the body before causing diseases or damage. These are two types, innate and adaptive immunity. These are the cells of innate and adaptive immunity. Rather, you can remember one thing, immune system is made, not born. Food and environment is the most important. So these are the points we can have to remember during considering our immunity. Now these are the pathogens responsible for diseases: prion, virus, bacteria, fungi, protozoa, parasites. And immune system is the third line of defense. First is the barrier. We have several barrier. Skin থেকে শুরু করে সব barrier, চট করে কেউ ঢুকতে পারছে না। Then level 2 innate immune system, some chemicals, নাকে থেকে শুরু করে যে ফুটোগুলো আছে সব কিছু না কিছু আছে, গার্ড দিচ্ছে, কেমন? And third is the adaptive blood warriors. These are the attacking invaders, some cells. Okay, so they have skin, mucous membrane, secretions, mucous secretion, phagocytic cell, antimicrobial protein, inflammatory response. And when you go for antibacterial compound from nature's na, most these things come, particularly antimicrobial proteins. Short proteins are there in many food. Even tomato, few days back there is a paper published in Microbial Ecology probably, and that paper told that tomato juice has got 2 peptides, and that is good antibacterial, particularly against typhoid. Okay, just few days back the paper has been published. So tissue and organ of this particular immunity system, that is immune system, one is bone marrow, another is bone marrow and thymus. These are the cells. Okay, now innate and adaptive component of immune system, these are the innate component: neutrophil, monocyte, eosinophil, basophil. Adaptive is B lymphocyte, NK T lymphocyte, and adaptive is cytotoxic T lymphocyte and helper T lymphocyte. Okay, and you see very very peculiar CD4, CD8. How many of you know the name? কতজন শুনেছো CD4, CD8? এরা কি করে? কখনো প্যাথোজেনকে হেল্প করে, কখনো হোস্টকে হেল্প করে। So they have duality, and this duality was discovered by Dr. Bhaskar Saha in its series of publication in Nature Medicine in 2004 to 2007. On the basis of that, he awarded the Bhatnagar Award. চট করে পায়নি, অনেক ফাইট করতে হয়েছে, but that was the remarkable work of duality of CD4. Okay. Now types of immunity: innate and acquired immunity. You see, these are the relationship: natural, artificial, natural, artificial, and artificial immunity by vaccination. Today I do not have enough time to teach you how the vaccine works and how after an infection what happens within the body. Each and every step, it's a very very interesting. Okay, but time limits is there, and this is the interplay between the two wings, and this interplay was damaged in coronavirus, in dengue virus, and that's why cytokine storm takes place. Storm means ঝড়। যখন cytokine কি হয়, innate immunity কাউকে ডিটেক্ট করার পর সেটা অ্যাডাপটিভ ইমিউনিটির কাছে পাঠায়, ও স্পেশালাইজড। এই পাঠানোর সময় যদি অ্যাডাপটিভ ইমিউনিটি ঠিক রেসপন্স না পায়, তাহলে ও বুঝতে পারে না। তখন বোথ ইনেট অ্যান্ড অ্যাডাপটিভ ইমিউন থেকে অনেকগুলো কেমিক্যাল সিরিজ অফ কেমিক্যাল সিক্রিট হয়, তারা একে অপরের এগেনস্টে কাজ করে। দ্যাট ইজ কল্ড cytokine storm. So these are the, actually see, innate immunity, lysis, blocking, a lot of things are there: interferon, deficins, lysozyme, if a virus attack or some bacteria attack, healing is there, but or an injury occurs, but there are different type of cells, they interact together with the complement system, and either they activate or they adhere, and the activation and presentation of these things in myeloid and other tissues, and then in adaptive immunity the specific things done. But if specificity loses, then only problem is there. বাসে দেখবেন চলতে চলতে অনেকে ছোট্ট একটা কথা বললে চট করে রেগে যায়, তার মানে আগে কোথাও ইরিটেটেড হয়েছে। বডির মধ্যে যদি কিছু ইরিটেটেড জিনিস ঢুকে থাকে, করোনাতে যে লোকগুলো মারা গেছে ভাইরাস মারেনি, কিন্তু ভাইরাস কাউকে মারতে পারেনি। কারণ একটা জিনিস আমি বলেছিলাম আমার টেলিগ্রাফে এবং টাইমস অফ ইন্ডিয়াতে আমার একটা সাক্ষাৎকার বেরিয়েছিল, একটা না সিরিজ অফ সাক্ষাৎকার বেরিয়েছে সেই সময়ে, সব নেটে পেয়ে যাবেন, ইয়েতে গুগল সার্চে গেলে পেয়ে যাবেন। আমাকে 3টে প্রশ্ন করেছিল। ফার্স্ট প্রশ্ন হচ্ছে, এটা হচ্ছে 2020 8 এপ্রিল, যখন আমি 24 ঘন্টাতে প্রায় 1.5 ঘন্টা আমি মানে ডেমনস্ট্রেট করেছিলাম জিনিসটা। আমি তখন ডাইরেক্টর, ওখান থেকে ডাইরেক্টলি কলকাতা থেকে সম্প্রচার হয় সারা ভারতবর্ষে। আমাকে প্রশ্ন করেছিল যে এই ভাইরাসটা কি করে এলো, এক। সেকেন্ড হচ্ছে কত লোককে ইনফেক্ট করবে। আমি বলেছিলাম 1 to 1.5%, 140 কোটিতে কত হয়, আর সেটাই হয়েছে। কতদিন থাকবে, আমি বলেছিলাম যে আমি তো জ্যোতিষ নই, কিন্তু আমি একটা জিনিস বলতে পারি, जो आया है उसको जाना पड़ेगा, आज नहीं तो कल। তাই হয়েছে। কেমন? আর ভাইরাস ভীষণ কিন্তু খুব রিজিড চরিত্রের, মানুষের মতো নয়। মানুষ কি করে, উপরে যে বসে থাকে সে নিচের লোককে শক্তি প্রদর্শন করে। ভাইরাস কিন্তু কখনো নিচের গ্রুপে কাউকে অ্যাটাক করতে পারে না, সব সময় উপরে অ্যাটাক করে। ওকে, and each virus has its distinct characteristic. For your information, the first definition of virus have been put forward by D. Chattopadhyay in 1999. Total consolidated definition of virus. You can consult any scientific paper, you will find it. Okay, so come back to the point. Human immune system evolved to deal with short-term threat, but not long-term threat. That is important. Unable to deal with long-term threat or ever-ending threat, which we now encounter every day as chronic stress, processed food, environmental toxin, and thus chronic inflammation contribute to a wide range of diseases. I have already mentioned these are the diseases. Quite is killing us, because modern research revealed that the link between inflammation and chronic disease is there. So cancer, dementia, depression, diabetes. Recently I got a project on dementia from Ayush. It's a good project, nearly 1 crore. Okay, we are going to see some of the very important parameters, and I will rather invite your young colleagues, young researchers, those who are doing very good work, actually those who have some time to think, we can join hand together. Okay, because I am still related with many of the Government of India organizations and Government of India funding agencies. Okay, so we can think on that. Now the counter disease of civilization, researchers are searching for equivalent of germ theory. Particularly microbiology students can recapitulate what is germ theory of 19th century. And during 90s to 2000, research demonstrated the role of inflammation in cancer and diabetes, and now extended into many other diseases. As the evidence continue to grow, the medical community covering on a unified theory of chronic disease based on the concept that chronic low-grade systemic inflammation is an underlying cause. You see what happened? When we take food, we do not know our bodily need, and even we do not know which food we are taking. Do you know? খাবারে কি আছে আমরা জানি, আর আমাদের বডি কি চাই তা জানি না। দুটোর মধ্যে কি হয়, একটা গ্যাপ তৈরি হয়, that is called nutrition gap. And if the gap continue, body sends some signal, and we unable to interpret those signals. Even if we go to doctors are trained for medicine, not for diet. Dietary scientists are trained with diet, but they do not have sufficient strength or sufficient laboratory facility to study each and every individual or a group of individual. So there is a long gap, huge gap, and continually when the nutrition gap is continuing, the body ultimately develops symptom to diseases. It goes to the genetic level and ultimately create a havoc. This is called the theory of disease causation. I have a book, Amazon-এ কিনতে পাওয়া যায়, খোঁজ নিয়ে দেখবেন, তাতে আমি খুব ক্লিয়ারলি ডেসক্রাইব করেছি যে হোয়াট ইজ দ্য মানে প্রসেস অফ ডিজিজ মেকিং, পার্টিকুলারলি লাইফস্টাইল ডিজিজ কি করে হচ্ছে ফুড থেকে। So now inflammation I have already told, and chronic inflammation, you see, persistent inflammation characterized by infiltration of lymphocyte and plasma cell, accumulation of macrophage and tissue destruction, attempt to repair but unable to do that, and they are classified non-specific, proliferative and granulomatous inflammation. Now these are the mechanism. You see, some of the body cells which are rather lymphocytes, specialized lymphocytes are responsible for these. I am not going in detail because you see these are actually picture. From this picture it is difficult until and unless you go inside it. But this is not a class to teach. This is actually the information. I am disseminating certain information in the capacity of a scientist, research scientist of global repute. Okay, and Elsevier as well as Stanford University list, you take 2% scientist list, you will get my name there. Okay. So pathway involved in chronic inflammation is JAK-STAT pathway, MAPK pathway, TLR signaling, NFKB pathway, and other pathways correlated together. These are the very established pathways so far known for the chronic inflammatory diseases. Okay. And chronic inflammation and atherosclerosis, I will show some example actually. How to build up plaque in the arteries. Okay. How arterial plaques are building. If you have omega-3 in your diet on a particularly say thrice in a week, development of plaque will be reduced. Only omega-3. If you do not believe, take few omega-3 tablets, and if you have an accident, puncture the tablet, puncture the content of the omega-3 fatty acid, put it there. Within 3 to 4 days, you will heal. Okay. So this is the healing power of omega-3. Omega-3 is a food, now it is used as a medicine. So food as a medicine. Then chronic inflammation and cancer. When DNA of normal cell altered or mutated, actually what happened, by birth very few people come with a defective system that ultimately leads to cancer formation. Majority of the people develop cancer after birth. How? Because of their DNA mutated some way. Mutation is a natural phenomenon. ও তো হবেই, আমার কাজ কি করে তাকে আটকানো যায়, কেমন? So when DNA of normal cell altered, the cell became cancerous or altered or mutated. Small percentage of people are born with genetic mutation that lead to cancer, but most people get cancer due to genetic mutation after their birth. Okay, so these are the chronic inflammation, alcohol consumption, carcinogen. You cannot prevent carcinogen. বাইরের বাতাসে যান 550 কেমিক্যালস আপনাকে স্ট্রাক করবে। আপনার আমার লিভারের মধ্যে P445 1 and 2 এনজাইম সিস্টেম আছে, they can detect, but eliminating huge amount of carcinogen by the liver is very difficult because our food is already adulterated. None of the food you will get good one. Okay, 20% related to chronic inflammation, 30% smoking and pollution, 35% obesity and dietary factor, and 15% is genetic predisposition or other factors. This is the classification. Now gene mutation may be rapid, may be failed to growth stop, altered DNA repair gene, and chronic inflammation may in healthy cells. Research found that reactive oxygen and nitrogen species produced during chronic stress induced inflammation that cause mutation. So inflammation to mutation, mutation to cancer. So, and this is type 2 diabetes. When diabetes, I think you all know what is diabetes. ওটাকে আর বলতে হবে না। এই যে সুগারটাকে আমি যখন খাবার খাচ্ছি সেটা ভাঙছে গ্লুকোজে, গ্লুকোজ যাচ্ছে ব্লাডে, ব্লাড থেকে ইনসুলিন গ্লুকোজকে নিয়ে কোষে কোষে ঢোকাচ্ছে। ওই কোষে যদি কোনো পর্দা মানে ঢুকবার জায়গায় যদি দরজাটা বন্ধ করা থাকে তাহলে তখন টাইপ 2 ডায়াবেটিস আসে, তাকে বলে ইনসুলিন রেজিস্ট্যান্স। ইনসুলিন রেজিস্ট্যান্ট কিন্তু 2 বছর টাইম দেয় রিভার্ট হবার, তা না হলে ফুল ব্লোন ডায়াবেটিসে যাবে। সো ইট ইজ রিভার্সেবল and it's a man-made disease. Diabetes is a man-made disease. The person is responsible. একই ফ্যামিলির দুটো ভাই দুজনে একই খাবার খাচ্ছে, একই সাথে বড় হয়েছে, একজনের ডায়াবেটিস হলো একজনের হলো না। শুধু খাবারকে দায়ী করলে হবে না, আরো তার সঙ্গে অন্য কিছু আছে কেমন সো you see body unable to use insulin well then it is called insulin resistance or unable to make enough insulin বেশিরভাগ লোকেরই সাফিসিয়েন্ট ইনসুলিন থাকে গ্লুকোজ বিল্ড আপ ইন দ্য ব্লাড and greatest risk is the body weight okay now high fat diet high carbohydrate diet is not so important but high fat diet is very important high fat diet মাংস খাবো তাতে তেল নেই তাহলে আর কিসের মাংস তাই না করি না আমরা তেলা মাংস খুঁজি কি জানো কি রিয়াজি পাঁঠা রিয়াজি খাসি inflammation relation what is the relation of inflammation with type 2 diabetes type 2 people have elevated pro-inflammatory cytokine inside fatty tissue thus obesity induced inflammation response contribute to inflammatory response or excess body fat trigger inflammation that build up cytokines in the fat tissue and leads to further inflammation type 2 diabetes inflammation contribute to inflammation inflammation contribute to type 2 diabetes so it's a vicious cycle okay 15.2 to 12.5% type 2 diabetes are not overweight normal weight but they got diabetes why because inflammation contribute to their diabetes internal slow level long term inflammation even doctor or that person do not understand but very simple symptoms are there that we neglect rather একটু ক্লান্ত লাগছে ও ঠিক আছে ঘুমোলেই ঠিক হয়ে যাবে সবাই সবার ডাক্তার আর পশ্চিম বাংলায় ডাক্তারের কোনো অভাব নেই রাস্তায় বেরোলেই আপনার ডাক্তারের সঙ্গে দেখা হবে যদিও তার ডিগ্রিটা নেই এটা আলাদা আলাদা ব্যাপার কিন্তু ডাক্তারই সে জানে ওষুধের দোকানে যান তারাও আপনাকে ওষুধ দিয়ে দেবে ডাক্তার দেখানোর দরকার নেই হ্যাঁ ওভার দ্য কাউন্টার ওষুধ পাওয়া যায় ওটিসি তো এইগুলো কিন্তু ভীষণ খারাপ করে কেমিক্যাল cigarette smoke injure cells that cause inflammation a significant risk factor for diabetes people with chronic inflammatory disease have a higher risk of type 2 diabetes about 1/3 chronic hepatitis C patient develop diabetes okay so now fibrosis and chronic inflammation leads to fibrosis এইটুকু জেনে রাখলেই হবে chronic inflammation leads to fibrosis these are the molecular pathway I'm not going in detail now pharmacological potential of selected phytonutrient আমাদের কয়েকটা কাজ এখানে দেখাবো আমি ডিটেইল বলবো না অ্যালিসিন কিসে পাওয়া যায় পেঁয়াজ আর রসুনে আমরা এটাকে জার্নাল অফ এথনোফার্মাকোলজি এবং আমাদের জেবিসিতে পাবলিকেশন আছে হ্যাঁ সেটা খুব কমপ্লেক্স ইভেন্ট বলে আমি এখানে সবটা আঁকিনি কেমন বার্জিনিন এটাও আমাদের আইসোলেট করা কম্পাউন্ড আমরা গাছ থেকে আইসোলেট করেছিলাম বহু খাবারে পাওয়া যায় এখানেও দেখুন আমাদের জার্নাল অফ বায়োলজিক্যাল কেমিস্ট্রিতে পাবলিকেশন আছে টিবি ম্যানেজমেন্টে কিভাবে কাজ করে এখানে কে বললেন যেন টিবিতে কাজ করেন we have several publication in a very recognized journal of the world okay very good work and probably you know the name of Dr. Govardhan Das he is also my collaborator and Ved Prakash Dwivedi my PhD student so we have and curcumin এটাও আমরা কাজ করেছি curcumin-এর উপরেও টিউমার সাপ্রেশনে curcumin প্রচুর কাজ হয়েছে পরে আমি বলবো কেন এগুলো ড্রাগে আসছে না পরে আসছে কেমন আমাদের সবার ধারণা আছে এগুলো বোধহয় ড্রাগ হয়ে যাবে না নেভার dietary components never regarded as drug okay so these are some of the এই তৌসিফ এখন আমেরিকার সিটিজেন ফ্রন্টিয়ার ইমিউনোলজিতে ওর এটা পাবলিকেশন আছে then inflammation and curcumin several membrane phospholipid to arachidonic acid how the things are synthesized and what curcumin did in this pathway then cancer and curcumin then luteolin another compound we have isolated it has multifunctional phytochemical then luteolin mediated TB immunity that prevents disease relapses okay then luteolin in anti-inflammatory potential then ginger a multifunctional phytonutrient it has also got a lot of and I am putting the documents evidence rather these are evidence based description or evidence based talk not like most of the people used to tell আদা খা ভালো হয়ে যাবে কেন কতটা ডোজে লাগবে you see the theme of establishing National Institute of Traditional Medicine of ICMR at Belgaum under my directorship was to validate these type of চিকিৎসা ব্যবস্থা, আমাদের যে দিদিমাদের যে চিকিৎসা ব্যবস্থা তাকে ভ্যালিডেট করা, এবং আমরা অনেকগুলোকে ভ্যালিডেট করেছি। আগামী দিনে বেশ কিছু মানে ওষুধ ফর্মে বাজারে আসবে, ফর্মুলেশন ফর্মে, নট অ্যাজ মেডিসিন বাট অ্যাজ এ ফুড, অর ইউ ক্যান সে ফুড সাপ্লিমেন্ট অলসো, বাট দে উইল বি ভেরি বেনিফিশিয়াল, দ্যাট মাচ আই ক্যান টেল, বিকজ রিসার্চ এভিডেন্স ফ্রম ডিফারেন্ট ডাইমেনশন। আমি অ্যাকচুয়ালি কাজ করি যখন না 3-4 ল্যাবরেটরি থেকে ডাটাকে সাপোর্ট করা হয়। আই হ্যাভ সাম ফরেন কোলাবোরেশন and those collaborators are very helpful of checking the things of us. So this is also gingerol, then resveratrol, then quercetin. You have heard the name, na? Impact of omega 3. You see, this is omega 3 actually, fish. You are talking about fish. So you see, EPA, eicosapentaenoic and docosahexaenoic acid. These two are not readily available in the body. We all know some plant omega 3 from plant, they are the precursor of omega 3, like ALA. ALA should go to the body through 26 steps breakdown, but many of the people do not have sufficient enzyme of these 26 steps breakdown process. So it's better to take easily available form of EPA and DHA, and that is available from some fishes, but you see those are sea fish, and sea is a mercury polluted area. Huge mercury pollution is in the sea. So you have to find out the exact composition or exact amount of EPA and DHA in as a supplementation. Anti-inflammatory, decrease platelet, then omega 3 harvested from salmon of deep cold ocean water is the best. নরওয়ে থেকে হয়, বেশ কিছুটা জায়গায় করছে এখন। নিউট্রিলাইট বলে একটা অর্গানাইজেশন কিছু করে, তাদেরগুলো কিন্তু সত্যিই ভালো। গত 25 বছর ধরে তারা কিন্তু সারা পৃথিবীতে রাজত্ব করছে, টপ। ওকে, দেন রেশিও অফ ওমেগা 6 এন্ড ওমেগা 3, সায়েন্টিস্টরা বলছে 4:1 হওয়ার কথা, আমাদের ডায়েটে 25:1। সো ইনফ্লামেশন হবে না কেন? অ্যান্ড omega 3 reduce the inflammation. কি করে করে? মেকানিজম আছে তার একটা। কি করে omega 3 ইনফ্লামেশনকে রিডিউস করে? Depletion of DHA, change in neurotransmitting receptor, and you see randomized double blind trial also there. Several trials are there. I have taken one. Okay. Now legume fruit, legume food rather you can say, nuts and many other things are there. They have protein complex, carbohydrates, starch, fiber, PUFA, phytosterol, vitamin and minerals. So these should be included in our diet. Now this is a plant from Andaman. We got several activity and these are the publications. Actually in a single plant we have always for making the plant in a in-depth study to validate the plant, we have done series of work, and that actually lead each and every plant 5 to 6 publications per plant, and good journal, phytotherapy, contraception, phytomedicine. These are very good journal, not very easy to get accepted from the paper from these journals. Similarly Malotus, another, this plant is from Andaman, and Adivasi use those things. Okay, then is Acaranthus, and even neem. We have isolated a new constituent that got antimicrobial and anti-HSV activity, okay, along with anti-inflammatory. Then Odina, this is from Jungle Mohol, work of Durbadal Ojha, who is now in National Institute of Health. Okay, this it has got Indian patent. You see 2 papers are there, PLOS 1 and J applied microbiology. Then you see Phyllanthus, it is another weed rather. We have several publications on it. Okay, US patent is there, and these are the synopsis of our work with 7-8 patent, 2 US patent and other Indian patent. And how to reduce inflammation by these things. How to reduce inflammation. You see, rest, ice and good wound care may relieve discomfort in few days for damaged things. Chronic inflammation, you need supplementation of, particularly in your diet, A, C, D, mineral like zinc, fish oil like omega 3, and some spices like garlic, ginger and turmeric. If you include in your diet particularly 2 to 3 times a week, you will get this relief from chronic inflammation for long time. Okay. Non-steroidal anti-inflammatory drug, there are several drugs, and steroidal injections are there. So 3 ways are there. You have to choose which one you will take. Doctors will go either second or third. But if you can convince the doctor, convince means you cannot convince anybody. You convey in a proper way. Doctor automatically convince. Okay. So synthetic inhibitor of inflammation, steroid and non-steroidal anti-inflammatory, their side effects are there. Several side effects on how they work. Okay. Now home remedy. What is the home remedy of inflammation? Eat more anti-inflammatory food. And it is your duty. I am talking about the persons, those who are working here. Take your own food. Go for analysis of its anti-inflammatory property. I will tell you the very developing model rather. You can develop a model. It's not very difficult. Develop an in-house model, test the inflammatory, anti-inflammatory property of the food and list it, send to ICMR. They will publish it. Okay. Then oily fish, eating too much of some food increase inflammation, like fried food, cured meat and nitrates, highly refined oil, refined carbohydrate. These are the things. Now today's world, it is very difficult to avoid many of the food, but you have to choose very selectively because you are in the outskirt of the so-called city. Okay. So obesity means obey the rule of city. I have written it in my book. Diet means either eat properly or die improperly. That is called diet. Vegetable is মানে ফ্রাই করে টেবিলে আনো তাকে বলবে ভেজিটেবল। আমরা যা ভাবি আর কি, vegetable is not green to us. Understand? So these are the many small things you have to consider. Now how to prevent inflammation? To decrease the risk of chronic inflammation, achieving maintenance weight, healthy weight, avoid or quit smoking, physical activity, rather break physical inactivity, limit alcohol intake, and manage stress with healthy tool. And these are the common dietary anti-inflammatory components. Foods are food, vegetables, phytochemicals, phytonutrient, and nutraceutical. Now major classes are carotenoid, flavonoid, lignan, isothiocyanate, phenols, saponin, sulfides with thiols and terpenes. These are carotenoids classification, you know. These are polyphenols, and these 5-6 concentrates: spinach, parsley, watercress, helencha, alfalfa, acerola, and carrot. It can mean for many of the inflammation can be controlled by these concentration. In our diet, what we take? Macromolecules, carbohydrate, protein, fat; micromolecules, vitamin and minerals; water; and another is phytofactor. That comes your vegetable, fruits, as well as your salad. How many of us take fruits and vegetables and salad on regular basis? Very few. I will tell you, the families in which some diseases are there, they will take care of. Otherwise, there will be a problem. Okay. Now these are the phenolic compounds, a list of phenolic compounds, a list of other groups also: carotenoids, monoterpene, and protease inhibitors. These are anti-inflammatory effect. Natural anti-inflammatory agents act in this way. It's a very complicated signal process, signaling pathway. Okay. And this is another pathway, regulatory network of TLR, in which we have worked. Two molecules we have worked with the TLR pathway. And this is a clinical trial. Several clinical trials have been conducted. I have presented some, not all, but if you go for the trial record, you will get a lot of trial. 4 slides are there on trial. And these are hypothetical model how phytochemicals work as an anti-inflammatory molecule to prevent your chronic inflammation. Okay. This is actually, you don't have to understand everything in a single lecture. You try to realize. I just here to aware about the facts. You may not have a garden in your premises, but you have seen garden, you have seen the green fields in your area. So encourage them and give some good words to them so that they will culture or cultivate all these things. Try to find out natural things as many as possible. benefits of phytonutrients. They are not essential for life but vital for good health. Remember this, essentiality and vitality is two different things. You can survive without this, but you cannot thrive without those. So nutritional role of phytonutrients: serve as antioxidant, enhance immunity and cell-to-cell communication, allow body defense to work efficiently, alter estrogen metabolism, cause cancer cell to die, repair DNA damage, detoxify carcinogen by stimulating cytochrome P450 phase 2 enzyme system. How phytos work with nutrients? How nutraceutical work, intercellular communication, endocrine system, and intracrine, autocrine, juxtacrine, these are the, and paracrine, 4 things. Okay, and as a neurotransmitter also. Now you see, this is a very classical picture, electron micrograph identifying a cancer cell large and spiky. Upon fishing with the cancer cell, the macrophage or smaller white cells inject toxin that kills the tumor. And here the ACM, same image, scanning electron micrograph of neutrophil, yellow color, engulfing the anthrax bacilli. Okay. Now cell signaling pathway I have already mentioned. You see, complex system of communication. When you are alone now, you talk with you, that is called self-talk. And when you are with others, you talk with others, that is called cross-talk. Each and every cell has self-talk and cross-talk. What input you are giving to self-talk, or what input self-talk যখন করছেন তখন কাকে বাঁশ দেবেন সেটা যদি ভাবেন তাহলে আপনিও বাঁশ খাবেন, বুঝলেন? কিন্তু সেলফ টকের জন্য যদি আপনি ভাবেন ওই ছেলেটার ভালো করতে হবে, ওর এই করতে হবে, আপনার কিন্তু ভালোই হবে। All are related to your body because mind cannot be dissected under microscope. Okay, but mind has a very powerful things because we have two mind, conscious and subconscious. আজকে যিনি ভালো কথা বলতে বলতে শাড়ি পরেন, সেই মেয়েটি ক্লাস 9 এ পড়ার সময় প্রথম শাড়ি পরেছিল, মানে সেফটি পিন, দড়ি, গিট ফিট দিয়ে কোনো ক্রমে ধরে যাচ্ছে যাতে খুলে না যায়। আজকে যিনি গাড়ি চালাতে কথা বলছেন, তিনি গাড়িকে ধাক্কা মেরেছিলেন যখন শিখেছিলেন, কনসিয়াস মাইন্ডে শিখেছে, সাবকনসিয়াস মাইন্ডে চালাচ্ছে। আপনি যদি কোনো জিনিস প্ল্যান করে সাবকনসিয়াস মাইন্ডে নিয়ে যান, আপনি সেই জায়গায় পৌঁছতে পারবেন। বাট ইট নিডস এ ট্রেনিং। সেই জন্য গুরু খুঁজতে হয়। আমরা গুরুর পরিবর্তে এখন গরু খুঁজি, সেটা করলে তো হবে না। হ্যাঁ, উই হ্যাভ টু সার্চ দ্য লিডার। এখানে যেমন প্রফেসর নন্দী রয়েছেন, কেশব রয়েছে, হ্যাঁ, এই রকম মানুষকে খুঁজে বের করতে হবে। হ্যাঁ, ability of cells to perceive and respond to microenvironment is the basis of repair. Okay, information processing error ultimately leads to autoimmunity, cancer and diabetes. And this is the importance of diet. Some as antioxidants, some as detoxified, some are modulating immune function. আর একটা জিনিস আপনাদের বলি, ইমিউনিটিকে বুস্ট করার কথা সবাই বলে। এখানে একজন এক্সপার্ট বসে আছেন, প্রফেসর অনল, তিনি হয়তো এগ্রি করবেন, হি উইল অলসো এগ্রি যে immunity cannot be boosted, immunity can be modulated. Immunity is not a single thing, it's a system. How do you boost a system? আপনি একা একটা সিস্টেমকে কি করে বুস্ট করবেন? So those are wrong words. আমি অনেকবার বলার চেষ্টা করেছি, কিন্তু কে কাকস্য পরিবেদনা, কে কার কথা শুনে। হ্যাঁ, মনে রাখবেন, আমি ছাত্রদের জন্য বলছি, ছাত্রছাত্রীদের জন্য, ইমিউনিটি বুস্ট শব্দটা ভুল। ইমিউনিটি মডুলেট করা যায়। হ্যাঁ, বুস্ট হচ্ছে যার মেন্টাল মেকআপ আছে তাকে বুস্ট করা যায়। যেটা হার্ড বায়োলজি থিংস, তাকে কি করে বুস্ট করবেন? তাকে ইরিটেটেড করতে পারেন, নট বুস্টিং। ইরিটেশন আর বুস্ট একই নাকি? আমি চিমটি কাটলাম, বুস্ট হবেন? ইরিটেটেড হবে। ভালো কথা বললে বুস্ট হবেন, ভালো কথা বললে মেন্টালি বুস্ট হবেন, ফিজিক্যালি নয়। কেমন? সো এ ওয়েক আপ কল, হাউ লং ডু ইউ এক্সপেক্ট টু লিভ? হোয়াট ইউর লাস্ট 20 years will look like? To evaluate healthcare, look at death rate of the country. Okay, quality of life, living longer or living better, which you want? Wants to live up to old age with Alzheimer's, cancer, cardiovascular disease, arthritis. These are the US figure. Or you have to say it like this. Okay, now conclusion: phytonutrient rich diet help to facilitate cell to cell communication, modify cellular uptake of hormones, vitamin conversion, repair DNA damage, detoxify carcinogen, supply antioxidant to prevent cancer, cause apoptosis of cancer cell, enhance immune response, prevent CVD, cardiovascular disease, osteoarthritis, osteoporosis, macular degeneration, cataract, and diet and supplementation should include a wide variety of whole plant food and whole food with broad spectrum of phytonutrients. So whatever I told is the research study, no claim of treatment. And these are my students who is actually the powerhouse. I am nothing. I have not done anything except managerial role. And to work with the students, still when I was the director, before the last day of my service, I was in the lab table to work in my own hand. And that publication comes in Frontiers in Medicine. Okay, I have done in my own hand. So I am indebted to all my students who did MD, who did PhD, who did postdoc with me, and as well as MSc and MTech I have not mentioned because list is too long. Okay, these are my students and all are well established. He is a principal of a college. She is in Germany. Sonali Das is also a professor of an university. She is also a professor. He is working in a government setup. She is in IISc. Ved Prakash is the group leader in ICGB. Then Anurup is also working in government sector. Durbadal is in US, which I am talking about. He is from Midnapur. And Day-1 is also in a publication house. Amit Kaur is in IBSD. And Yashpant Masure is working in a company. And Vicky is also in US. Ashok is also in US. Neelam is establishing her own company. Arti is in US. Ananya is in a university. And Joy Mondal is in government service. And Reva Thakur just submitted his PhD, already got actually. So he is moving to US, and Rubika is under submission. Okay. And Nidhi is in Delhi University. Annesha is working in a government fund. Chiranjit is in a college, and Priyanka is also in a university. So, and these are those who help in this slide preparation. Dr. Joyita Ghosh, a nutritionist and dietitian, Amity University. Dr. S. Simran from Indian Institute of Science, Indian Institute of Statistical Institute, sorry, Indian Statistical Institute, and Navonita is from Cancer Research Center, Kolkata. So I am grateful to all these people. Now this is a big thank you for all of you. Have I finished in time? Sir, time maintain hua kya? Thank you, sir. Thank you. And I mentioned two things, sir.
Nicely presented.
Your director and your principal and vice principal, as well as Dr. Pradhan. I have gone through their biodata, and I have seen they have done tremendous work, and these young minds should be. That is true. I have seen their biodata and some of their publications. Thank you so much, sir.
And on behalf of the organizing committee, we are very much grateful to you for your nice presentation, and even also and patience, particularly for the younger students. And there are so many dynamic faculty, energetic faculty are present here, including the supervisors and the others. And intensively he has been discussed about the information along the causes, the reason, the symptoms, even also how to improve or the treatment. Very nice strategy and practical experience. This is much more important. And may I ask the questions from among the students, mainly the students. Any questions? Otherwise, the speaker
এই লজ্জা পাওয়ার
is
দরকার নেই,
disappointed.
কোশ্চেন করতে পারেন।
কেউ করতে কোশ্চেন
যার যা মনে আছে, কারণ
বাংলা, ইংলিশ, হিন্দি,
যে কোনো ভাষায়। এই চারটে ভাষা জানি আমি।
কানাড় যদি জানে, নিশ্চয়ই কাজ করেছেন, কর্ণাটকের ভাষাও জানেন।
বুঝি আর বলতে পারি না, বুঝি।
আচ্ছা, বুঝেন, তাহলে ঠিক আছে। Anyone? Yeah, you may. First your name and your affiliation institute. নাম অফ দ্য, তোমার নাম বলবে আর তুমি কোথা থেকে আসছো বাবা। নাক থেকে একটু বন্ধ করো, এখন আর কোনো ভাইরাস নেই। হ্যাঁ, খুলো তোমার মুখটা একটু ভালো করে দেখি আমরা সবাই। আমি বেশি পড়ি, হ্যাঁ বেশ সুন্দর, ঠিক আছে বলো। নাইস।
আমার নাম দীপ
এই এই
তুমি সামনে চলে এসো বাবা।
না, অন্যরা শুনতে পাবে না আবার।
ও হ্যাঁ, তার মাইক্রোফোন
yeah,
লাগবে।
yeah.
এটা নিয়ে।
Hello, my name is Deep Chattoraj
ওকে,
and I am from Shantiniketan, Bolpur.
বাহ, ভেরি
I'm
গুড।
studying here at NUD Nutrition and Dietetics PG first sem. So, sir, basically, while I was listening to your presentation, I just had a question. Just when you came to the part of insulin role, sir, I just wanted to know, there are two types of hormone. Cortisol is also affecting hormone. We have seen people regularly leading to stressful life, for which the cortisol production is increasing day by day. Can this be a cause to the insulin factor in our body?
Sure.
Can I know anything about it?
It is actually what happened. Every hormone, there are two different hormone, one opposes other. Insulin, similarly for insulin, when something steroidal things are introduced in the body for long term, body adapted with that, and a number of other hormones cannot able to prepare in that proportion. So their inability or activity, rather lose activity. In steroidal condition, if you introduce steroid in a human body, all the steroid cannot be eliminated easily. It takes time to eliminate. So body by this time changes its mechanism. So it is reported actually. It has report. And you are right.
So, sir, I just wanted to know one more thing. That is, whether it is antagonistic to the insulin or is it
Yes,
effective?
it is antagonistic. Right.
Thank you, sir. That's all I wanted to know.
Okay. Anything more?
এনিথিং মোর
ওদের একটা হাত
অ্যামংস ফ্রম দ্য, হ্যাঁ কে হাত তুলেছে?
দাঁড়িয়ে পড়ুন।
ইওর নেম?
Good afternoon, sir. My name is Anushree Patro, studying in PG section in this college in zoology.
গুড।
Sir, my question is, as a habitant of urban area, how can we avoid the adulteration-free food? I mean, how can we adapt the adulteration-free food?
Very nice question. Actually, adulteration is a man-made thing and it harbor in our mind. Short term-এ বেশি লাভ করতে চাই, তার জন্য adulterated, এবং আমরা আমাদের ছেলে মেয়েদেরও adulterated করে দিচ্ছি. We are adulterating our মানে next generation also by demanding a lot of things. তোকে এই হতে হবে, ওই হতে হবে. That is also adulteration. One thing you have to do, you have to do your work whether they are listening or not. আজ নাই তো কাল শুনেগা। তোমার কাজ বলে যাওয়া যে এইটা করলে তোমার পরিবারে কি ক্ষতি হবে। এই কোভিডের সময়ে আমি একটা স্টেট অফ আর্ট ল্যাবরেটরি তৈরি করেছিলাম and I was felicitated by the Karnataka government. First time in India, ICMR এর কোনো সায়েন্টিস্ট বা ডিরেক্টরকে রাজ্য সরকার বড় হল ঘরে সমস্ত টিভি ফিভি সবকে নিয়ে এসে আমাকে সে ইয়ে করেছিল, ফেলিসিটেট করেছিল এটার উপর বিরাট ব্রোঞ্জ দিয়ে এর উপর দিয়ে কি বলে একটা স্ট্যাচু দিয়ে। তো কারণটা হচ্ছে আমি ওদেরকে শেখাতাম যে ভাইরাস থেকে বাঁচবে কি করে। শুনতো কি না শুনতো সেটা আমার কথা নয়, আমার কাজ হচ্ছে বলে যাওয়া। তুমি কিন্তু রাতারাতি কিছু করতে পারবে না বাবা, তোমার কাজ হচ্ছে অ্যাওয়ারনেসটাকে ক্রিয়েট করা। কে কি বললো ভুলে যাও। আমি আইএএস আইপিএসদের পড়াতাম এক্স্যাক্টলি, প্রতি সপ্তাহে একটা করে ক্লাস নিতাম ওপেন জায়গায়, সেখানে আইএএস আইপিএসরা বসতেন। আই হ্যাভ এ লিস্ট অফ মানে গ্যালাক্সি অফ আইএএস আইপিএস অফিসার আমার বন্ধু। কর্ণাটকে যান, যে কোনো জায়গায় আমি ফ্রিতে ব্যবস্থা করে দেবো, জাস্ট সিঙ্গেল ফোন কল। দে রেসপেক্টেড মি লাইক এনিথিং। হ্যাঁ, কারণটা একটাই, বিকজ তাদেরকে আমি ভয় ভাঙিয়েছিলাম। তাই এটাই বলি, আমরা সব সময় বলে যাই, আমি তো জানি শুনবে কি শুনবে না। হ্যাঁ, 80-20 প্রিন্সিপাল, ওকে, প্যারোটিস প্রিন্সিপাল বলে। আওয়ার ডিউটি ইজ টু ডিসিমিনেট। আর তুমি ইয়াং স্টুডেন্ট যখন এই কথা বলবে না, ওরা আস্তে আস্তে বুঝবে কেন বলছে, তার কোনো স্বার্থ নেই। বুঝতে পারছো, নিঃস্বার্থভাবে কিছু বললে কাজ হবে, একদিনে হবে না। ওকে বোঝাও এই জিনিসটা কি তুমি খাও, যদি বলে না আমার জন্য আলাদা চাষ হয়, তাহলে বলো তোমার ছেলে ছেলে আর ওদের ছেলেগুলো ছেলে নয়। সেন্টু দিতে হবে কখনো কখনো, কারণ যার সেন্টিমেন্ট নেই তাকে তো সেন্টু দিতেই হবে, তাই না?
থ্যাংক ইউ,
ভালো কোশ্চেন করেছো,
থ্যাংক ইউ।
Thank you, sir.
তুমি কি
Thank you.
উত্তর পেলে? কারণ এই উত্তরটা পুরোটা আমার জানাও নেই, আমি তোমাকে যা বলছি সেটা আমি যা করি তাই। ওকে,
Okay, awareness is the first thing that we have to do first.
শিওর,
Thank
তোমাকে তোমার কাজ করতে হবে। নেক্সট,
ওকে
আর যারা যদি কিছু
ওকে,
থাকে,
বেটার হচ্ছে আপনার যদি কোনো ইয়ে থাকে তাহলে কোশ্চেন করে নেবেন, এবং উনি অনেকক্ষণ রয়েছেন,
আমি আছি আজকে রাত্রিটা
আজকে
আছি,
রাত্রি
কারো
আছে,
কোনো প্রশ্ন থাকলে করতে
দেন
পারো।
এনি টাইম ইউ পার্সোনালি মিট উইথ দেম,
Thank you, sir.
মিট উইথ হিম।
Thank you,
ওকে,
sir.
নেক্সট।
থ্যাংক ইউ সো মাচ।
Okay. We are very much grateful to you, particularly for that.
এ তো ভাই
Very nice. It is a continuous process. It is not a discuss within a 1 hour. Within a 1 hour. খুব দারুণ। You have a experience. Professor Chattopadhyay, you are most experienced in down to the earth. Even also lab to the land. This is the most important thing. Yeah, okay, thank you. And next speaker, I am very much proud for him. Actually, last 4 decades, more than 40 years, is connections with a great Professor Keshav Chandra Mandal. Since the undergraduate from Midnapore College to my colleague as a student in the year 1985 and 86, then as a colleague, it is very much another proudfulness of the Professor Keshav Chandra Mandal. After that, even also the researchers in also advise many times then how to do and how to enrich yourself. And I learned so many things, my students. I have a lot of students in the level of school level, even also the college level, university level, whatever may be. I lot of things learn from them. And particularly in the year of 1985, what I actually you know, I am now excited also. Actually, the 4 musketeers, the Professor Keshav Chandra Mandal, in the professors of the Vidyasagar University now. Another one is the Jodhpur AIMS, Dr. Professor Paschim Chandra Nayak. He is also another student, and that time in the Midnapore. The another one is the University of Calcutta, the Professor Nilkanta Chakraborty. Another one is so many, but very important role played in my life that time. When I teach them, they ask so many things within my bedroom also, in the evening, any times, the morning to evening. That's the trigger of myself. Then I learn so many things, my students. This is very much important. And even also my research scholars and the others. Not a few things about the Keshav Mandal. Keshav Mandal, as a teacher, is a good teacher, and very perfect and innovative teachers. And as a researcher, and also the supervising in the nice way, in innovative way. And another thing is social behavior. Very much interesting, social behavior to the society. Then how to how to move for the society, have a good society. This is very much important role played by the Keshav Chandra Mandal. This is, I am very much proud now, till now, and till associations with the Keshav Chandra Mandal and his family, as because his wife also my students. And that time I have and so many students are present here, and as a faculty, as a students, and also the researcher. So I am very much proud, and particularly for Keshav Chandra Mandal. Okay. Keshav Chandra Mandal, wait. Keshav Chandra Mandal does start his journey as a student, then as a lecturer. That time is the lecturer, not assistant professor, like that type of the designation, not in offered by the government or the others level. In the College of Raja Narendra Khan Women's College, in the Department of the Human Physiology, and after that he moved to for the research in Vidyasagar University in the microbiology department, and very founder microbiology department was in Professor B.R. Poti, and only the two faculties are established a lot of things in the department of the microbiology, and his product is till in front of me as a Dr. Kuntal Ghosh, and so many, and the others, faculty also present here, Subhasis and the others. And after that, he also published lot of papers, already mentioned Professor Sarkar, lot of papers, lot of book chapters, and also awarded so many award, particularly very limited in mention here, and gold medal as a first rank in the Vidyasagar University, as because Keshav Mandal and that time that all the students who are taught in Vidyasagar University in from the initiations of the university in the year 1985, mostly, no? 85 or 87, like that. Okay. Then, and got the first rank. And even also the obtained the Ramendra Sundar Sinha Memorial Award 2012 from Physiological Society of India. Then also obtained the awards, the Fellow of the Biotech Research Society in India, and also he has handled so many major, minor, and the innovative projects amounting. Hello, kichu holo সমস্যা? ভাই কিছু সমস্যা হয়েছে? তোমরা চলে যাচ্ছ কেন? আবার আসবে তো? চলে এসো। নেক্সট হচ্ছে উনি হ্যান্ডেল করেছেন প্রায় 1 কোটির উপর, 2 কোটির কাছাকাছি, from the department, so many department of the government of India, and also guided the students, lot of students, more than 16 already awarded, thesis submitted 1, and registers still now in the 5, and MSc and others, MPhil dissertations, and so many. I do not mention in details. So may I request Dr. Keshav Chandra Mandal to deliver your speech in this topic. Okay.
Thank you, sir,
Okay.
for your nice word. Jan ebare bosun. So, firstly, I offer my sincere gratitude, regards to my Gurudev, the excellent teacher, the particularly the organizer who invited me in this seminar. There are one of the eminent scientists, Debuda, is here, and after his lecture, it is very tough to say anything. So believe me, I will conclude within 30 minutes, whatever he mentioned. And another my friend, Dr. Anol, and all the principal and other staff and my beloved students. So now the concept is being famous, vocal for local. Okay. We have developed civilization for more than 6,000 years. And we have a cultural heritage, food cultural heritage. what sort of lacuna we are facing now in respect to adulteration or other things, that so-called highlighted not more than 50 years, within the last 50 years. But if you believe our culture, believe our food culture, simply for example, if you look after the biryani handi, okay, or the Lay's kurkure like the material, just you see the energy content of this material. As you are from the nutrient background, so just see in case of 100 gram of kurkure, it has written, well written, the quantity of energy more than 500 calorie. So these are so-called nutrient poor, energy dense food. There is no nutrient. Just you consume 3 to 4 packet, your energy balance will be maintained. That is the major cause of the lifestyle disease. So I particularly highlight the so-called saturated fatty acid, the all sort of devastation mostly made by the saturated fatty acid. This is the major cause of all sort of disease. So beside the nutrient, whatever mentioned Debuda, many phytochemical, many potential nutraceuticals, I will focus another part of the body which is remain hidden. People not look after this part. This is called gut microbes. If you consider a adult human, we have around 10 to the power 13 number of cell, eukaryotic cell. But only within the gut, that containing more than 10 to the power 15 number of cell. Only in the gut. So within our body, simply we can say that we are rather prokaryotes than the eukaryotes. So these group of so-called microbes is now highlighted when from the 2006 gut microbiome project initiated, 2006. And now people look after, and many, because this is a diverse, that composition varied from region to region, food culture to food culture, and many factors are associated. But they remain in a standard ecological system. When their composition changes, that is called dysbiosis. Dysbiosis, that means when the ecological balance of the gut flora changes, that is called dysbiosis. And what Debuda mentioned, the chronic inflammation, that theory somehow changed now. Now theory comes the leaky gut concept. So chronic inflammation when happen, that due to many causes, or I can say that your food actually, food composition actually, for example, you are consuming saturated fatty acid containing material, saturated fatty acid containing food. So what will be its impact? So initially this fatty acid will be inflamed the gut mucosa. Then change happen with the so-called epithelial layer, leads to changes of the permeability of the gut mucosa. And through gut mucosa, endotoxin of the microbes diffuse into the body. And that leads to chronic inflammation. That thing happen. Many diseases are now linked with the changes of gut microbial dysbiosis. Okay, many diseases, so-called diabetes too, obesity too, and many diseases. and the mood, depression, that sort of neurological behavioral change are now linked with the gut microbiota dysbiosis. Initially, we have the idea that hypothalamus pituitary axis is the central axis of the body. Now the concept has been changed, gut-brain axis. This is the major axis considered by the many people. So, within the gut, many microbes are there, but among them one group of microbes, fungi also there. Many bacteria are there, many different phyla, many groups are there. So if you consider, they are so teeny, therefore the weight of the 10 to the power 15 number is 1 to 2 kilo. But 95% of the bacteria, and these are their genome, if you consider the genome, within, perhaps you have idea, from 1993 to 2003, one initiative started that is called Human Genome Project. Then people have the idea that if you sequence the gene, then we can control the all disease, all sort of disease. But after analysis, from the 2005-6, many literature come, and where it has established that we have strong 25,000 gene, remaining 70,000 gene are called junk gene. Then people highlight how our homeostasis maintained, how our cellular function maintained. For that reason, new initiative started that is called Gut Microbiome Project from 2006. And now the people are linking the activity of the microbial genome with the host genome. There is a link, co-expression system. Microbial genome can regulate expression of the host genome. So what I stated earlier, prokaryote highlight. So, or if you consider the functionality of human, okay, then not only the human, every animal, all animal are the superorganism, as because this is the byproduct of both prokaryote and eukaryotic cell. So, among the gut microbiome, some are always harmful. These groups are always harmful. And majority of the group which are responsible for the gut dysbiosis are called facultative group. Next, some green color, these are always good, healthy. Okay. So now focused this good flora as probiotics. So probiotics mean some group of bacteria having always exert health beneficial effect. There are many definitions, but here two important definitions that World Health Organization or ISAPP more or less same. Live organism when consumed in adequate amount confer a health benefits to the host. So using this, just you see market opportunity, huge market opportunity. Everywhere you can get probiotic tablet to control the disease. For many consequences, this is one sort of natural therapeutics. In its growth rate around 7.9%, and if you consider the probiotic yeast, so one group is the bacterial yeast, another is the probiotic yeast. Okay. The market of the probiotic yeast is around 5.3%, as I will highlight some example efficacy of the probiotic yeast. So just you imagine its rate, growth rate all around the world. So these are the major player of the probiotic bacteria and probiotic yeast. Majority are the probiotic bacteria. That means these are always confer health benefits to the host. So among them, one Saccharomyces group has been considered though it is a yeast, but it exhibits health promoting activity. So if you compare the difference between two groups, one is the bacteria and yeast probiotic. If you compare, as because in the market there are two types of probiotics are available. For human, non-human, huge number of probiotics available. If you compare their efficiency, that these points like facilitate the absorption of nutrient, both positive yeast and bacteria, helps break down undigested carbohydrate. You know that our host system, for example, in case of human, we have very limited number of carbohydrate degrading enzyme. We have very limited number of carbohydrate degrading enzyme. But with our food, we consume different types of carbohydrate. It has estimated that gut flora able to secrete more than 1,000 types of carbohydrate degrading enzymes. Simply it has estimated that 30% of total energy, 30% of our total energy, for example, normally 2400, okay, it's 30% looks like the 800 like that, that quantity of energy extracted by the gut flora. Extracted means that food portion digested, breakdown by the gut flora, 30%. So both bacteria and yeast helps with digestion, helps maintain balance of healthy intestinal bacteria, both maintain what I mentioned earlier, support overall immune system, both bacteria and yeast. Next come, as it has larger size, so increase coverage and intestinal protection. So yeast probiotic, this portion is the yeast, and this is the bacteria. Just you see the comparison. So as these are the larger size, so that can protect the intestine. Number one, this is the very important. Continues to work during antibiotic use. So whenever we consuming any antibiotic, that can destroy bacteria, all sort of bacteria. These are bacteria specific, but yeast are non-responsive to antibiotic, so they will be escaped. Okay. Next, withstand the harsh environment. Withstand the harsh environment. So whenever we consume or remain what mentioned that stressful or other condition, various condition, in that condition bacteria can't survive, but yeast can survive there. Reaches to colon in an active state. Only the yeast, majority of the yeast, stimulate production of IgA. It appropriate for the age 2 months and up. What thing happen if you see the dynamics of the gut flora? Dynamics of the gut flora, it change during the stages of life. Okay, but yeast flora is somehow constant. They are somehow constant. So therefore, many product are in the market, are maintained using the yeast. Okay. So if you compare, for example, normally whenever we suffered by any gastric problem, we consume the antibiotics. This is a comparative list. You just see whenever we consume the antibiotics, what thing happen? It will degrade the pathogenic organism, not only the pathogenic organism. Suppose within this room, there are some good bacteria, some pathogen, or majority are the somehow non-reactive. But whenever we consume the antibiotic, that destroy the all bacteria, irrespective to good or bad. Then what thing will happen? Leads to antibiotic associated diarrhea, Clostridium difficile infection. That is very, very important. mineral nutrition related diarrhea. So that after consuming of the antibiotic leads to antibiotic associated diarrhea. But if you consume, for example, in the market there are many yeast tablet, yeast probiotic. So that not only prevent the growth of the pathogen, but also stimulate the immunity. That is the major plus point of the yeast, that stimulate the immunity. Whenever immune system becomes strong, so both end or in two way, one directly reduce the growth of pathogen and other way stimulate the immunity, then it can easily cure the gastric problem, stabilize stool consistency. Though the antibiotic treatment is rapid, but probiotic yeast treatment is sustained and health supportive. So there are many benefits of the probiotic. I am not going into the details. So why probiotic yeast is healthier than the so-called probiotic bacteria? One of the point I already mentioned, that immunostimulatory. Okay. In the yeast wall, there are a component called beta-glucan. This is the cell wall of yeast and fungi. One of the component is the beta-glucan. That beta-glucan actually induce the activity of macrophage, T regulatory cell, therefore enhance the secretion of the interleukin. This is the simple science. Okay. Just you imagine, within the gut there are trillion number of bacteria. So how whatever we generally mention, that probiotic and other important thing. 1 minute. AC টা চালিয়ে দে তো, গরম লাগছে। So, what I am mentioning, that this beta-glucan, the communicator, just you imagine the communicator between gut and the health. Just imagine our intestinal lumen. Okay, this is in one side. In the lumen there is bacteria. Bacteria unable to cross the barrier. So how gut flora or how probiotic exerted their beneficial effect? This is one question, as they remain within the gut. So major connector, mediator between the two sides. Major mediator between the two sides. One is the gut, another is the host cell, is the microbial metabolite. One of the thing. That is now the priority of the research. 2-3 mediator have been identified. 2-3 mediator, one is very common, is the short chain fatty acid. Short chain fatty acid. There are many choline derivative, many amino acid derivative. These are the communicator between the gut and the host cell. Okay. So what thing happened? This intestinal wall, within the intestinal mucosa, there are some immunosensitive cell. They have some finger-like projection into the lumen. They are acts as a sensor of the intestinal environment. Am I clear? Within the intestinal lumen is quite different. Within the lumen, there are some projection of the dendritic cell. They actually sense the environment, that means particularly they stimulated by the beta-glucan and leads to activation of the T regulatory cell and other immunological cell, and enhance the immune power. This is the basic thing. Okay. So now come to our experiment. In this presentation, I am stating very brief. So our aim is that we have explored our traditional one yeast, which consuming our people with their traditional drink in our locality. So this yeast consumption is not a new one. From the ancient civilization, there is an example of the leavened bread. In our all sort of baked bread containing the yeast. But before that, traditionally people harvest the yeast, not only for the production of wine or ethanol-like product, but that used as a food ingredient, okay, from the ancient time. So we have isolated one yeast, then studied its morphology and its behavioral pattern, its biochemical activity. For studying any microbes, we have to need identifying them. We have to study their biochemical characteristics, and we have identified them as a Saccharomyces 1, Saccharomyces cerevisiae. Okay, this Saccharomyces cerevisiae is the prominent group in the marketed probiotic yeast. Saccharomyces boulardii, Saccharomyces cerevisiae, these are very close to each other. They are 99% genome are similar. Okay, we have isolated one Saccharomyces on the basis of biochemical and molecular characterization. And next we have studied their probiotic efficiency. Those who have the idea about the probiotic or working in this field, we have to test some biochemical principle, whether these are supportive to the organism or not. Simply it looks like that. Just you imagine our gut environment. First phase is the acidic, second phase is the alkaline, and containing one dangerous component, bile. Okay, so for establishing one organism, we have to study their stability at different pH, gastric juice, and many harsh environment. If they cross, then we will designate this as a good probiotic one, good supportive one. But after that, you must have to test for a particular disease, considering the definition of the probiotic. Okay, you have to consume adequate amount, and that confer a health benefits. So characteristics like that, these are the characteristics. Stability at the acidic condition, bile, alpha mileage, these are the some characteristics, and this is the score. It somehow crossed the limit score. Therefore, it is possible to demonstrate that this organism could cross the intestinal barrier. Next, we have studied the animal experiment. And for our motto is that whenever we consuming any product, that is first react with the gastric mucosa. Okay, so very common now the acidity, gastric ulcer, many things. These are the what Debuda stated earlier, chronic inflammation of the stomach particularly. Okay, so we have studied whether this probiotic is preventive to gastric ulcer, and for this we have used the animal and then grouped into three groups, one normal, one ulcer, and another treated with our desired yeast. Three group. Okay, and next we have studied their so-called histological structure. And this model is based on the sudden cold treatment. This is one established model. And we have observed that the group 1, this is the control group, and this is the yeast treated group, is quite similar. In case of so-called ulcerative group, just you see the ulcer point and the histological change. So these are proved one of the evidence that these can protective to the gastric ulcer. So next we have studied some ulcer index like factor, and by calculating this, it has established that our studied organism is it can inhibit the ulcer, its rate is around 84.13. This is some sort of method we have employed. Next question arises, as these are so-called inflammatory changes, then I am simply stating these are called pro-inflammatory cytokines. Always balance. It should be balanced between pro-inflammatory and anti-inflammatory cytokines. These are commonly designated as the pro-inflammatory, these are the anti-inflammatory cytokines. Okay. Next we have examined the content of the inflammatory cytokines. As these are the immunological consequence, so we have observed that our studied yeast, what I mentioned earlier, it can, as because we applied the yeast previously before the treatment, that induce supportive, preventive for gastric ulcer, as it can reduce the concentration of the pro-inflammatory cytokines, and on the other hand, upregulated the expression of anti-inflammatory cytokines. Okay, so this is the simple presentation of my research. So thing is like that. So this common yeast, it has enough potentiality as a immunomodulator, positively express, or we can say that it can support the gastric health. Okay. Now, my dear student, I want to mention few words what I initiated, that these are not the new thing, but these things occupied huge market. So there is huge possibility. You like people, young people, if you initiate some sort of entrepreneurship, what Debuda mentioned, many spice. Just you imagine, in our nature, in our forest, there are many plants, but among them, our ancestors selected few one, which we are now using as a spice. So this yeast we are cultivating from the ancient time. So now the time is that if you young people initiate something, government is helping us many support, startup like thing. This is a huge possibility. Every institute now popularizing the concept startup, not only depend upon the government job or like that. There are many components in your surrounding. Just try, just initiate. That will be better for you, your livelihood, as well as also the country. Country is now looking on your activity. Last night I exchanged the so-called fees of our government-sponsored institute with the Honal sir. You can imagine how government is supporting for you. So now the time You have to do something for the country, not for the country. There are enough possibility. So local food, just Debuda mentioned that the juice, that means tomato juice having anti-inflammatory component. Okay. And most of the tomato juice in our local market are not made up with the tomato. Okay. Made up with the pumpkin or like other thing added color. But in our very close, I visited that place particularly like Ajodha Hill, Purulia. There produce huge quantity of tomato. Huge quantity of tomato. And they are selling into the market 2 to 5 rupees per kilogram. So just there is enough possibility to get the natural product. This is also your duty to prevent adulteration. You are always depend upon the marketed spice, turmeric or any other flour. But you can initiate one industry here by using the pure turmeric flour. These are not a hard job. So there is huge, not depend upon only the service. Service sector is now so-called gradually declining number of service points. Just you perhaps read some news from the West Bengal. And yesterday only, government reduced the budgetary allotment for the education, as because many private institutes are blooming. Therefore, government reduced. This is the real scenario. So, with this, I am not mentioning details, but there is huge scope. Huge scope. In my teaching career, I am somehow grateful that I produce around 11 entrepreneurs. One of them is now so-called, his turnover more than 5 crore per year. All are selling the bacteria. All are involved in the selling of bacteria, not other thing. Huge scope is there. But you young people, you have to think that we can do everything. Okay, with this word, I thanks to the organizer one and my students for doing research. Thank you very much.
Again, formally thanks to our, hello, my student, please keep silent. And very interestingly presented by our Professor Keshav Chandra Mandal. And take-home message is very nice. You have to establish own of your own to establish any company and like that. This is very much important. And obviously, I asked the open for the session regarding the question and among from the students. After that, the faculties and the scholar. Any student? If you memorizedly heard it, then you have to question. Otherwise, not at all. Anyone? Yeah, you may. You are the only, the students present here, the listens attentively and seriously and mindfulnessly, so that you may much more congratulation and also the clappings, all the hands for him.
Sir, just now I heard about fermentation, something you were speaking about. I just want to know a small thing. That is, sir, when it's milk and let's suppose I'm talking about tartaric acid, what we get from imli or the other products. So why is it supposed to curdle rather than to form any casein form of it? Not spice. I'm saying about imli. That is.
Imli only.
Yeah.
Like the tartaric
Yeah. Acid thing.
acid.
I'm asking.
Acid coagulate the protein simply.
Simple.
But sir, on account of side.
That thing happen normally. Just you see, whenever lactic acid bacteria grown, that produce acid that coagulate the protein. Okay. There are many things in our tradition. I'm
चालू कर देना.
Huh. I'm pointing out. I saw in the particularly Sundarban zone, they use dried chili, Sugno Lanka as a starter. Okay, as a starter for production of doi. So microbes is needed. Microbes is needed for the production of doi. Okay.
Now microbes are present in the chili. Very nice technology you have learned, and that will be implemented in the near future. Okay. Okay. Yeah, good.
Just you see, in case of one of people also completed PhD on the Hania. In the many times, this is a one base drink.
Yeah.
Alcoholic, mild alcoholic, rice fermented, alcoholic beverage, oily, health supportive, contain multi-nutrient, multi-species bacteria. Okay. So I want to point out they not added any old starter for fermenting the rice. They use some plant residue for fermenting the rice. Next question arises, from where microbes come? Microbes, as I mentioned that within our body microbes are there. Like in case of plant, there are microbes are also there. These are called endophytic microbes. Okay. They reach from the plant residue for fermenting the rice fermented beverage.
Sir, lastly, I would like to ask another question. And the question is, we are often familiar with antioxidant intake, right? From fermented foods.
Okay.
Antioxidant rich we are considering. So, sir, basically, when it comes to form of alcoholic beverages,
Yeah. Alcoholic beverage.
yeah, let's suppose
This is
rhododendron.
Okay. This is highly anti-nutrient or other factor. No need to think about the anti-nutrient, anti-obesity like that. Just take your local food. All local food are healthy. All traditional foods are healthy. That contain all sort of nutraceuticals. Don't think like other thing. Just in this environment. So whenever you grown in this environment, nature support you like that. All sort of supporting thing also grown in your surrounding. Okay. For example, we are remain in the Gangetic plateau where huge raining, lots of ponds are here. You not find this sort of pond in the Uttar Pradesh or like other state. Okay. So you take your local fish that is contain what the stated omega-3 fatty acid. So in case of fish, majority of the omega-3 or unsaturated fatty acid remain in the head portion. But in our market, its value is low than the body. Similarly, in case of chicken, okay, majority of the so-called omega fatty acid remain in the breast, that means middle portion. But your choice is the leg piece. Okay, that is the real thing.
Okay.
Thank you, sir.
Okay, I think better to hand over to our coordinator, Department of English faculties. Is there any formalities for this session, technical session? Dr. Raj, even also pursuing PhD, Abhishek Chakraborty. Thank you everyone for cooperations and very much listening.
It was a mesmerizing session indeed. Thanks to all our eminent speakers for this wonderful session, and special thanks to Dr. Dilip Kumar Nandi sir for chairing it. Thank you so much, sir.
Now it's time for our lunch break. After lunch break, we will again return to the seminar hall to the Young Scientist Aware program. In the meantime, the free communication session will be held in the seminar hall, and it will be chaired by Professor Nirmal Kumar Sarkar. So again, we will meet here after the lunch break. Thank you, everybody, and thank you to our delegates. The Young Scientist Awards program will start from 3:30 p.m. in the seminar hall.
After lunch, we are requesting to come back here, all of you.
Now we are going to initiate the Young Scientist Award session.
I would like to request Professor Nirmal Kumar Sarkar to come to the stage and chair the session. Sir, please come to the stage. Now, sir, will be introducing Dr. Kaushik Das, sir, who will be presenting the paper. At the same time, I am requesting our madams to felicitate Dr. Kaushik Das, sir. Sir, please come on the stage. A big round of applause as our madams are felicitating Dr. Kaushik Das, sir. Again, a big round of claps, everybody. Now I'm handing over the session over to NK, sir. Sir, please take over.
Hello.
We are now beginning Young Scientist Award program, and today a single lecture will be included in this program. The speaker is Dr. Kaushik Das. It is customary that I introduce the speaker to you all before he starts his deliberation. Dr. Kaushik Das is Assistant Professor, cum HOD, Department of Nutrition of Belda College, Midnapore. Formerly, he was Assistant Professor in Raja N.L. Khan Women's College. Dr. Das is a young and energetic teacher as well as researcher. He has already supervised 4 projects funded by UGC and DST, Government of West Bengal. At present, 5 research scholars are carrying out their PhD pursuit under the supervision of Dr. Das. Dr. Das had been the organizing secretary of as many as 5 national level seminars held at different places. And he also acted as the keynote speaker in 2 seminars. He has presented his papers in a number of seminars, conferences, etc. He is quite young enough right now, but nevertheless, he has as many as 35 publications in different peer-reviewed journals. And quite importantly, he has already been the owner of a few awards, like Professor S.N. Professor Sochidananda Memorial Award from Physiological Society of India, Swarna Padak Experimental Nutrition Award from Indian Dietetic Association, and Presidential Award from Indian Dietetic Association. On behalf of everybody in this hall, I welcome Dr. Kaushik Das for presenting his paper.
Good afternoon, Dr. Nirmal sir, and good afternoon, everybody. Respected my sir, Dr. Dilip Kumar Nandi, respected director sir, Dr. Pradip Ghosh, and others dignitaries, and my dear student. Today, I will deliver my one research topic, the antidiabetic effects of Volavetsky-Cephas protein hydrolysate on experimentally induced type 2 diabetes mellitus in rat model. We all know about that. The diabetes mellitus. Diabetes mellitus is a complex disease characteristic by a chronic hyperglycemia, high blood glucose, due to deficiency of insulin secretion from pancreatic beta cell or resistant to bind with insulin receptor in skeletal muscle or fat cells. Insulin mainly bind with only the skeletal muscle and fat cell. When you are inactive, the skeletal muscle is inactive, then the fat cell uptake the glucose by the action of the insulin. So nowadays the sedentary lifestyle and various type of fast food, junk food consumption enhance the high fructose sugar and enhance the also the inactivity or physical inactivity. So skeletal muscle physical inactivity increases the glucose uptake in the fat cell, and fat cell is increased, and thereby the insulin receptor is increasing the fat cell, and insulin receptor decrease in the skeletal muscle cell. So nowadays 1 in 10 people are living with diabetes as per International Diabetic Federation Virtual Congress 2023 held on 7 December 2023 at Belgium. I am also member of IDF, and I am the online participant in the 7 December. Now 537 million people live with diabetes in the world. Type 2 diabetes mellitus is 90%, which is called the T2DM, 90% among all diabetes. T2DM or type 2 diabetes mellitus or high blood glucose causes 10 years shorter life expectancy, 2 to 4 times the risk of cardiovascular disease, 20-fold increase the lower limb amputation and kidney failure. At present, therapeutic management for T2DM patient uses 5 types of oral hypoglycemic agent, that is the diabetic medicine. For example, sulfonylurea, glibenclamide, biguanide, metformin, thiazolidine, pioglitazone, DPP4 inhibitor, gliptin, SGL2 inhibitor. These are the popular anti-diabetic medicine prescribed by the diabetologist. So these oral hypoglycemic agent or popular diabetic medicine, first is metformin. It is just a first type of screening when pre-diabetic stage. The doctor prescribe the metformin. Actually, this metformin is the mechanism of action is inhibit the gluconeogenesis and glycogenolysis. That means the reduces the fasting blood glucose level. And thiazolidine is the activating the nuclear receptor PPR gamma, peroxisome proliferate active receptor gamma, that actually this receptor increases the insulin sensitivity in the skeletal muscle. When increasing the insulin, this gene activation, then insulin can bind with the skeletal muscle. Glibenclamide inhibit the ATP sensitive potassium channel in the pancreatic beta cell, that decrease the insulin secretion. And now this just 10 years in the market, gliptin inhibit the DPP4 dipeptidyl protease 4 enzyme present mainly in the pancreatic beta cell and expressed in the pancreatic beta cell in when the pancreatic beta cell or we are the stress condition, and then enhance the GLP-1. Gliptin is the DPP4 inhibitor. DPP4 enzyme actually inhibit the GLP-1 glucagon-like peptide incretin hormone secretion from the intestinal cell when we load the food. Now this glyphosine medicine now in the market last 5 years, they inhibit the SGLT2 and increase the glucose secretion through urine. all the oral hypoglycemic agent work there via this type of separate mode of action, and ultimately reduces the insulin and increases the insulin and reduces the blood glucose level. So OHA or oral hypoglycemic agent many side effects: weight gain, hypoglycemia, nausea, skin rashes, liver disease, heart failure, shorter lifespan. Therefore, there is an urgent need to discover safer and natural alternative active ingredient without toxicity adverse effect to tackle this disorder. So by this the mechanism, we so many researchers and scientists continuously searching the food derived alternative therapeutic anti-diabetic agent with low or no side effect. Enzymatic hydrolysis of sea fish mass, we selected some sea fishes for their anti-diabetic activity. So we collected the sea fishes and it prepared the protein hydrolysate, sea fish protein hydrolysate, bioactive fish protein hydrolysate. Fish protein hydrolysate contain various type of bioactive peptides and protein fragment, scientifically proved as an antioxidant, immunomodulatory, anti-diabetic, anti-inflammatory, antibacterial, anti-cancer, and anti-hepatic effect. So some review literature on the fish protein hydrolysate and its anti-diabetic activity. The protein hydrolysate from herring, mackerel, salmon, and bonito fish improve the whole body insulin sensitivity. This is a reference. Atlantic salmon skill gelatin hydrolysate, DP4 inhibition in plasma is the reference review literature study. Halibut, tilapia skin gelatin hydrolysate, in vitro DP4 inhibition 38 to 51% inhibition by the feeding of the gelatin hydrolysate of the halibut or tilapia skin protein hydrolysate. Salmon fish protein hydrolysate decrease the postprandial glucose in rat. Bora fish muscle protein hydrolysate enhance the GLP-1 secretion and inhibit the DP4 activity is the literature study. So my study or our study was comprising by following aim and objectives. In our earlier study, it has been proved that lower prevalency of type 2 diabetes mellitus among sea fish eater than freshwater fish eater. Literature study suggested that bioactive fish derived peptide are crucial source of anti-diabetic agent. So objective to fulfill the aim to prevent and manage of T2DM, we have selected a sea fish and designed to create protease generated sea fish protein hydrolysate to prove an anti-T2DM FPH against streptozotocin induced T2DM rat model and find out the threshold dose of fish protein hydrolysate as alternative therapeutic agent of T2DM. So why have I chosen the Bolavetki sea fish to identify the type 2 diabetes FPH, that is fish protein hydrolysate? Our earlier study has been revealed that sea fish eater consume more Bolavetki sea fish than other sea fishes. Also Bolavetki sea fish oil contain high DHA, EPA and plays anti-diabetic activity. Before this study or the any study, any research project, any research work when start by a problem, and that problem created and searching the solution create a research work. So problem is the type 2 diabetes mellitus and searching the solution is the type 2 diabetes mellitus, how it will be prevented by a natural food product. So we previous this study we around 2020 before corona we studied the Shankarpur Digha Mohana on the sea fish eater, mainly sea fish eater, huge amount of sea fish daily consumed by the Shankarpur Mohana Digha Mohana area people, and also Kamarpukur and Bolpur area people, they do not consume any sea fish, and the blood glucose level we collected from those people, those region of people, and we can found that sea fish eater Shankarpur area people are lower prevalency T2DM than the Bolpur, Kamarpukur area people. So it is hypothesized from this study it may be sea fish consumption and related with the type 2 diabetes or blood glucose level reduction. So from this point we collected the CPs, and those people, Sankurpur area people, huge amount of Bhola Bhetki CPs consumed, daily Bhola Bhetki CPs consumed, because Bhola Bhetki CPs, they are only 10 rupees per kg. So very low cost. So Bhola Bhetki CPs, first the Bhola Bhetki CPs, we prepared the Bhola Bhetki protein hydrolyzate by following methodology: collection of Bhola Bhetki CPs, clean properly, fine chopped, 50 milligram per 1 HBSS solution, homogenization, and 10 milligram protease enzyme with homogeneous mass solution, incubate, and then by the following thoroughly processed, we prepared the purified protein hydrolyzate from the Bhola Bhetki CPs. And peptide sequencing and determining the amino acid composition by from this Bhola Bhetki CPs protein. This is the methodology, second methodology. Estimation of fasting blood glucose and glycosylated hemoglobin concentration, because when the any research on the diabetes mellitus, first is the criteria, the main parameters or biosensors is the fasting blood glucose and hemoglobin A1C, that is the glycosylated hemoglobin. Measure the glycosylated hemoglobin, estimation insulin and C-peptide concentration in plasma. Why we measure the C-peptide? Because the insulin is secreted from the beta cell, and C-peptide is produced by the breakdown of the proinsulin. So one C-peptide molecule means one insulin molecule is secreted from the beta cell. So it actually insulin and C-peptide measured because it is the type 2 diabetes. Is it the type 2 diabetes mellitus or type 1 diabetes mellitus? Because the various literatures said that the streptozotoxin, we actually induce the diabetes mellitus by the streptozotoxin, a drug. Various many researchers demand that the streptozotoxin injection induce the type 1 diabetes, but we just prove by the C-peptide concentration measurement, actually in the type 1 diabetes there is no insulin in the blood, so there is no C-peptide in the blood. But when insulin is secreted, within 1 minute insulin bind with the receptors. So insulin sometimes is not a sensor for the type 1 or type 2 diabetes, but C-peptide is not utilized by with our body. C-peptide is just metabolized and excreted through our body. So C-peptide measurement is a hallmark for the type 2 diabetes mellitus. So estimation of glucagon-like peptide, GLP-1, dipeptidyl protease DPP4 activity in pancreatic tissue. Because the GLP-1, when we consume the food, the pancreatic beta cell signaled by the GLP-1, glucagon-like peptide 1, it is secreted from the intestinal L cell. Because when glucose load in the intestine, GLP-1 secreted from the intestine, and it GLP-1 bind with the beta cell GLP-1 binding receptor GLP-1R and signal the insulin secretion. But DPP4, dipeptidyl peptidase 4 enzyme, it breaks the GLP-1 when the pre-diabetic or stress condition in the pancreatic beta cell. Next parameter, evaluation of the activity of the antioxidant enzyme. Because the diabetes mellitus is the main cause of the diabetes mellitus is stress condition in the pancreas, stress condition in the body, and oxidative stress actually main culprit for the increasing the hyperglycemia. So this is the biosensor of antioxidant enzyme, catalase, SOD, glutathione by the our biochemistry biochemical method. Evaluation of lipid peroxidation and histological preparation, statistical analysis, and then immunohistochemical staining using DCPH2 and to determine the ROS level, oxidative stress level in the pancreatic beta cell. This is the methodology. And cell apoptosis assay by the Beckman-Coulter flow cytometer, and these are the experimental design. experimental when we collected the eight groups of rat designed and 5 rats per group. First control, vehicle control. Control is the fed the normal food and water. Vehicle control fed the normal food water. Diabetic streptozotocin administration with high lipid diet and water for 28 days. And diabetic with FPH, that is a fish protein hydrolyzate consumption, is the 4 group. Is a dose dependent threshold dose to identify the threshold dose, so dose dependent manner. FPH 125 milligram per kg body weight, FPH 250 milligram per kg body weight, 500 milligram per kg body weight, and 750 milligram per kg body weight. And also a control diabetic is OH, that means the diabetic anti-diabetic medicine, also a group created. And glibenclamide 12 milligram per kg body weight we fed for 28 days, resulting the blood glucose when streptozotocin induced streptozotocin inject in the rat just after 72 hours. When we measure the blood glucose level, the normal control normal rat those are not induced by the diabetes, 80 milligram average blood glucose level per DL of the blood. But when after 72 hour injection of the streptozotocin, 600 near about 500 to 600 milligram blood glucose rising, hyperglycemia, severe hyperglycemia. That is approved by 0 day, first day it is a all group of rat, the normal below the 100 milligram per DL blood glucose. When streptozotocin injection here, then blood glucose level high, but surprisingly the fish protein hydrolyzate supplementation rat blood glucose level fall down. And also hemoglobin A1C also increase the blood glucose level, and also hemoglobin A1C the corresponding the increase the hemoglobin A1C. And this is the hemoglobin A1C, all the control group, the normal 4.5 or below 6 gram % of the HbA1c. But in the diabetic group, that is the diabetic group, the high above 10 gram % hemoglobin A1c. But when we supplementation of the fish protein hydrolyzate, reduction of the hemoglobin A1c. And also the insulin level, also the insulin control group, the insulin level, diabetic group insulin level decreased significantly. And FPH 500 milligram per kg body weight FPH supplementation showed the best result for the insulin and also C-peptide and GLP-1. Here the control GLP-1 is similar to the diabetic and fish protein hydrolyzate supplementation. And also DPP4 enzyme activity, there the control DPP4 enzyme activity, but diabetic rat, in the diabetic rat DPP4 enzyme activity is significantly higher than the control and vehicle control. But in the fish protein hydrolyzate supplementation, the varieties of the reduction of the DPP4 activity and highest reduction activity in the dose of 500 milligram per kg body weight. And also glucose 6-phosphatase activity is the enzyme, this glucose 6-phosphatase enzyme mainly the main biosensor of the liver. When diabetes or hyperglycemia occur, glucose 6-phosphatase activity expression is steeply high and increases the fasting blood glucose level by enhancing the gluconeogenesis and glycogenolysis. So this enzyme is a important enzymatic biosensor for a diabetes management model. And control, vehicle control, diabetes model glucose 6-phosphatase activity tremendous high, but in the supplementation of the fish protein hydrolyzate is the glucose 6-phosphatase activity is significantly reduced. And also glucokinase activity. What is the importance of the glucokinase activity measurement? Because the glucokinase is the enzyme when we consume the food, glucose first broken down in the digestive enzyme and absorbed to bloodstream, and when the glucose enters in the beta cell, first glucokinase আমরা কি জানি যে হেক্সোকাইনেজ হচ্ছে গ্লাইকোলাইসিস করে, বাট ইন দ্য বিটা সেল গ্লুকোকাইনেজ এনজাইম start the glycolysis, and by the after glycolysis TCA cycle and ATP is generated, and that ATP induced the insulin secretion. So high activity of glucokinase activity, that means the lower the chance of diabetes, that means the hypoglycemia. So glucokinase in the control rat is the higher than the diabetic T2DM rat, but supplementation of the fish protein hydrolysate increases the glucokinase activity, that means insulin secretion activity to ROT2. And also another superoxide dismutase, the antioxidant enzyme, catalase, it is also antioxidant enzyme, and GSH. These three antioxidant enzymes jointly work to reduce the superoxides. So here the superoxide dismutase in the diabetic rat, superoxide dismutase is lower, diabetic rat catalase is lower, GSH diabetic rat lower, but in the control group the significantly higher the SOD, catalase and GSH, but supplementation of the fish protein hydrolysate increases the SOD, catalase and GSH activity. But in the reverse mechanism of the MDA, malondialdehyde, that is the lipid peroxidation. This lipid peroxidation means the oxidative stress marker, direct marker in the beta cell, that when the diabetic rat, the MDA level is high, significantly high than the control, but in the MDA level is decreased significantly, the supplementation of the fish protein hydrolysate, and highest decrease significantly in the 500 milligram fish protein hydrolysate. It is the detection of intracellular reactive ROS measurement by DCHF2 staining in the pancreas. It is clearly shown that the control, vehicle control, there is no oxidative stress, but diabetes the stress, and FPH 250, 125 is the 500 is the best short. And cell apoptosis assay, here actually the apoptosis measurement in the pancreatic beta cell, pancreatic cells, here the control group and vehicle control group, diabetic group, the necrosis is high, but here the 125 and 250 and also the 500 and 750. Another detection of apoptosis by acridine orange and ethidium bromide staining in pancreas were done at here the control group, the cell apoptosis and necrosis, this diabetic group, and also actually these imaging methodology was proved or confirmed to the another and histological study. The control group of rat, the beta cell islets of Langerhans the normal, here the vehicle control normal, but here diabetic is the reduce the size, and diabetic fish protein hydrolysate 500, the normal, they are the normal the size of the pancreatic islets. And peptide sequencing of FPH, which prepared by in our laboratory FPH, peptide sequencing, here the low molecular weight some peptides found, low molecular weight peptide found. And finally the mechanistic mechanism of the FPH, we just hypothesized or conclusion the induction of type 2 diabetes mellitus by streptozotocin and Volavetki CP's protein hydrolysate feeding or supplementation. That peptide polypeptide, actually the biopeptide, it may be bind here, then DPP4 inhibition and insulin secretion, also GLP intestine, GLP glucagon-like peptide increase the insulin secretion by the beta cell. It is a proposed diagram of the mechanistic way, and it is now we are going, or we will be just a in silico model, just not a proved for the further proved for the this mechanism. So Volavetki CP's protein hydrolysate FPH experimentally proved as an anti T2DM oral hypoglycemic agent, food derived OHA, which reduces blood glucose level via DPP4 inhibition and GLP-1 agonist by increasing insulin secretion in rat model. Further, it will be required more study to identify particular anti-T2DM peptides through in silico analysis and rat model, and next with human clinical trial for establishment of new OHA in future. We this study is the supplementation of the fish protein hydrolysate, and further in my future work it will be assumed to biopeptides. We actually try to identify the anti-diabetic peptide that may be reduced the hyperglycemia, or a novel anti-diabetic peptide, a just comparative or compared to the anti-diabetic medicine OHA. A new OHA it may be, if we will success on the anti-diabetic identification or discovery of the anti-diabetic peptide, then it will be a new oral hypoglycemic agent. And the references, and these are the my research scholars. 3 research scholars already awarded PhD degree, and now 4 research scholars are running this work. Thank you.
Thank you very much, Dr. Das, for your beautiful presentation on the prospective potential of fish protein hydrolysate in management of type 2 diabetes. Now the lecture is open for discussion, and I think you don't mind if I begin the question answer session with a very silly question from myself.
Okay, sir.
As a layman, I'm asking a question. Do you think that cooked bhola bhetki fish will give some dividend, give some benefit to type 2 diabetes patients? Instead of preparing so many, passing through so many steps for preparing the hydrolysate.
Sir, it was also my question.
Will we get any benefit in diabetes, whether type 2 diabetes patients will get any benefit if they take bhola bhetki regularly and they cannot take raw flesh? Cooked fish will give any benefit or not?
Sir, during my discussion on the aim and objectives, where I already told that why bhola bhetki should choose? Choose because we already surveyed in a population model. শঙ্করপুর এলাকা, স্যার অ্যাকচুয়ালি শঙ্করপুর এলাকায় প্রচুর, মানে ওরা খুব গরিব, মানে শুধু লঞ্চে যায় আর কি, সব মানে ওই মাছ ধরতে যায়, ঠিক আছে, এবং লঞ্চে চলে গেল প্রায় ধরুন 1 মাস, কিছু টাকা দিয়ে গেল ঘরে, আর ওখানে যে যারা লোকাল জেলে শুধু ওই দেখি ভোলা, রুলি এইগুলো সব প্রচুর মানে ডেলি খায়, এবং আমরাও তো ওখানকার ছেলে, আমরাও মানে ডেলি ভোলা, ওখানে রুই কাতলা বেশি পাওয়া যেত না, এখন ওই ময়না নারায়ণগড় হয়ে রুই কাতলা আসছে বাজারে, আগে ওই বিলাসপুরের রুই উপর ভরসা থাকতো, অথবা আমরা ওই সামুদ্রিক মাছ খেতাম। তো ওখানকার এলাকায় দেখি শঙ্করপুর ওই এলাকায় আমরা 127 জনের উপরে দেখেছি, এবং হাউস ওয়াইফ দেখেছি, কারণ কোনো হাউস ম্যান কেউ ছিল না, কারণ সবাই চলে যেত মাছ ধরতে। হাউস ওয়াইফের উপর দেখেছি তাদের মধ্যে মাত্র 3 জনের ব্লাড গ্লুকোজ লেভেল হাই ছিল। বাট সেই কম্পারেটিভে আমরা বোলপুর কামারপুকুর এলাকায় 127 জন ফিমেলের উপরে দেখেছি, হাউস ওয়াইফের উপরে। তারা কিন্তু কোনো কাজ, শুধু হাউস ওয়াইফ জাস্ট। তাদের মধ্যে 45 মানে ইয়ে হাউস ওয়াইফের কিন্তু হাই ব্লাড গ্লুকোজ ছিল, এবং তার মধ্যে আবার 22 হচ্ছে আননোন ছিল। মানে আমরা গিয়েই প্রথমে আবিষ্কার করলাম যে ওদের ব্লাড গ্লুকোজ হাই, ওরা জানতেও না, বলছে আমাদের তো কোনো প্রবলেম হয় না, আমরা তো ফাস্টিং ব্লাড গ্লুকোজ নিয়েছি আর কি। এইভাবে দেখা গেল যে এইখান থেকে আমাদের রিসার্চটা স্টার্ট হলো যে তাহলে নিশ্চয়ই সামুদ্রিক মাছ যারা খায় তার নিশ্চয়ই তাদের কিছু একটা বেনিফিট হয়। সেই হিসাবে আবার গেলাম ওখানে যে ওখানে কোন মাছটা বেশি খায়। দেখি সব থেকে বেশি খায় ভোলা ভেটকি, তেল তাপড়া, তারপরে রুলি, এইসব মাছ বেশি খায়। তো সেখান থেকে আমি এটা দেখলাম যে ভোলা ভেটকি মাছটাই, তাহলে আমি দেখি এর ভেতরেই সব দেখবো যে ভোলা ভেটকির অয়েলে আমার একজনের পিএইচডি কমপ্লিট হয়েছে, 2টো পেপারও আছে, রিসার্চ পেপার। আর ভোলা ভেটকির অয়েল, এবং ভোলা ভেটকি তো খাচ্ছে বলে তো ওদের ব্লাড গ্লুকোজটা কম আছে। কারণ সত্যি কথা বলতে কি আমারও এখন ব্লাড গ্লুকোজ লেভেল হচ্ছে এই সেদিনকে মেপেছিলাম 86 মিলিগ্রাম%, কিন্তু আমার একটু কিন্তু প্রি ডায়াবেটিক চলে গেছিলো কিছুদিন আগে। আমি মানে ধরুন করোনার আগে আমার প্রায় 120-25 থাকতো। আমি এখন ডেলি এখন সামুদ্রিক মাছ খাই, রাত্রিবেলায় দিনের বেলায়, ঠিক আছে। মোটামুটি এখন মনে হচ্ছে যে আমারও ব্লাড মানে জাস্ট এ ওন হিউম্যান ট্রায়াল। না, হ্যাঁ রান্না করেই তো খাচ্ছি।
তাহলে একটু বারেও ভালো, কাজও বেশি হয়।
মানে একটা গ্রুপে হচ্ছে ডায়াবেটিস অ্যান্টি ডায়াবেটিকের বদলে একটা কুক। অয়েল নয় স্যার, ফিশ প্রোটিন হাইড্রোলাইজেড।
মানে বয়েলিং পার্ট ছিল, এইবারে বয়েল করলে ওর কোনো কম্পোজিশনে
অ্যাকচুয়ালি কী হয় আমাদের তো, আমরা যখন খাচ্ছি আমাদের তো যে এনজাইমগুলো আছে যেগুলো প্রোটিয়েজ এনজাইম, মেনি থাউজেন্ডস অফ প্রোটিন এনজাইম প্রেজেন্ট ইন দ্য ইন্টেস্টিন অ্যাক্ট অন দ্য প্রোটিন। তো দে ব্রেক্স দ্য ভেরিয়াস পেপটাইডস, বাট মানে উই just try to break down by particular 3 peptides: alkalase, protease and pepsin, and they just cut the peptide a particular sequence, and then that peptide we just feeding মানে
একটা বয়েলড গ্রুপকে অ্যাড করুন, দেখুন আপনার কি মানে কতটা হিট হবে দেখুন। যদি এটা পাবলিশ তো হবেই,
কুকড গ্রুপ পার।
ইয়েস, এটা একটা রিমার্কেবল কাজ হবে কিন্তু।
হ্যাঁ, থ্যাংক ইউ স্যার, এটা খুব ভালো
সাজেশন থেকে যোগ করে ফেলুন, খুব ভালো কাজ।
Now I wish if some of the students could ask some question.
সেটাও দেখতে পারবেন।
Any question from any student?
পেপটাইডের পরিমাণটাও দেখতে পারবেন। আপনার তো বেশি
এক সেকেন্ড আগে ছাত্র কেউ বলে বুঝিয়ে। Any question from any student?
Kaushik da, thank you for your nice presentation. I missed some introductory part. Anyway, I want to ask one thing, that last you have shown the peptide sequences, right? So how you got this peptide sequences? From the multi-peaks you have identified the peptide sequences, or you have digested purposefully by any particular type of peptidase and you got it? Or if you kindly say something about it. Yeah.
Actually peptide sequences মানে we when we prepared the protein hydrolyzed just we send the lab.
Okay, so they have analyzed
and
they find out some sequences.
Find out the these the peptide sequences.
Yeah, so those are the huge number of 128 sequences, right? Amino acids length.
हाँ,
Okay, got it.
multi.
Okay, okay.
multi, MS and multi. And then this এগুলো সব access মানে সবই
But
found
which one have the highest score, or which one have matched with your species? That will be the relevant, I guess.
না
So-
এগুলো হচ্ছে অ্যাকচুয়ালি এতে প্রায় 250 থেকে 300 পেপটাইড ওরা দিয়েছিল, যেগুলো হাইয়েস্ট স্কোর সেগুলোই আমি দিয়েছি
No, topmost highest score.
Topmost
with
highest
your
score এগুলো।
species.
হুম।
Then that will be your protein, I guess.
ও ওইগুলো অত করিনি।
Any
Okay,
more
thank you.
question from anybody else?
আমি স্যার এই যে এখন একটা পেপার কমিউনিকেট করেছি, যেটা হচ্ছে লিখেইছি টাইটেলটাই যে দীঘা মোহনা ভোলা ভেটকি। হ্যাঁ স্যার, খুব ভালো সাজেশন এটা।
Now I would like to add a few sentences. I see Midnapore City College has grown quite rich. They will be presenting a Young Scientist Award to every participant in this program. So naturally, Dr. Kaushik Das will be getting a Young Scientist Award. So let's give a very big applause to Dr. Kaushik Das to encourage him to work more in the coming days. And one more thing, I wish I could also be young and participate in this.
You are young, just 24 years old, rest is your experience.
Sir, last year also I got Young Scientist Award. This year got Young
Young
Scientist.
plus young, you will grow old.
And it will be long to young or old age and then old scientist.
Young grade 2.
Grade 2.
Now I invite Dr. Devaprasad Chattopadhyay, invited scientist in this conference, and Dr. Pradeep Ghosh, Honorable Director of this institute, to kindly hand over the Young Scientist Award to Dr. Das. And I request the photographers to take a snap.
স্যার আপনি এদিকটা আরে আসুন না।
ঠিক আছে ঠিক আছে, আমি এখানে পেছনেই থাকা ভালো।
না আপনি মাঝখানে থাকুন আর আপনি এখানে বসুন, সুবিধা হবে সেটাই, তাই না নাকি? বসে নেওয়া যাবে না? না কিছু কিছু কনফারেন্সে বসে দেয়, কিন্তু হ্যাঁ আমাকে অনেকবার ওই করেছে মাঝখানে বসিয়ে মাথায় একটু পরিয়ে দিলে।
কিন্তু এইখানে একটু দেখতে ভালো হবে প্রাইসটা। আপনারা দুজনে মিলে কাইন্ডলি, দুজনে মিলে
ধরুন আপনি।
And some, I think some cash is there.
Congratulations and wish for more good work. Okay, continue this thing because দেখুন এইরকম জায়গা থেকে এই টাইপের কাজই তো চাই।
হ্যাঁ, লোকাল
এই কাজটার গুরুত্বটা আপনি এখন কতটা বুঝছেন জানি না, কিন্তু আমরা যারা সিনিয়র লেভেলে আছি
রিসোর্সকে এক্সপ্লয়েট না করতে পারলে
এক্স্যাক্টলি আমি ফান্ডিং এজেন্সিগুলোতে আছি, ডিপিটি বলুন, ডিএসটি বলুন, এরও আমরা চাই সিপিসের উপরে কাজ, ওরও সিপিসের উপরে কাজ। আপনি কি কোনো প্রজেক্ট দেন ডিপিটি ডিএসটি নতুন কলেজে গিয়ে কোনো প্রজেক্ট পাঠাইনি, এই টাইপের ভীষণ এনকারেজ প্রজেক্ট রেডি করেও পাঠানো হয় না, কারণ কলেজের একটু পলিটিক্যাল প্রবলেম। কিছু কাজে না একজন ডাক্তার সঙ্গে রাখে, হ্যাঁ এগুলো ছোট ছোট জিনিস। এটা যখন ডিএসটির একটা প্রজেক্ট ছিল স্যার, ডব্লিউপি ডিএসটি, সেখানে আমি প্রেজেন্টেশন দিতে গেছিলাম, আমরা বলি না ওরা খুব ভালো গ্রুপ ছিল, এখন চেঞ্জ হয়ে গেছে, ভালো গ্রুপ ছিল, আমারও ছিল একটা ডিএসটি।
Now it is time again to hand over the microphone to Sri Abhishek Chakraborty sir.
Now it's time for a little break, a tree break, and after that, our students will perform. They have organized a cultural program, so it will start from 5 p.m. So after the break, again, we will meet here in the seminar hall for the cultural program. Thank you, everybody.
Sonia Patro and I take the privilege of accompanying this event. I will very honored to have this with us respected professors, guest teachers and faculties. I welcome you all on this cultural program which is full of performance of the student of our college. So at the very beginning, I would like to start this event with a wonderful act performance. By this performance, today we are going to explain you the role of a woman in Sanskrit Nari, story of the one who hold our planet Mother Earth in their mighty hands. But what do they get in return? The creative hands, the creator, are now tied with the heavy chains of burden. The people who pass it off in disgust of responsibility should be strictly prohibited to roam freely in their own world. They are none by the woman from, from whom we all created, and not just as but the entire world is created. Now these women are sold commodities in our so-called civilized society. Do you know what is the most painful to one woman? A touch that she never wants to be touched, but it happens to them every day, every time, which creates fear, and the path of fear which leads fertility. Who will tell them the right path? Who will tell them how to overcome the fear? Who will tell them the light? From the Hindu holy book of Manusmriti, quoted by Lord Sri Krishna in Darpa Yug, Yatra Narayastu Pujayanti Ramante Tatra Devata. That means a woman as a Narayani. If she respected and worship for her duties, it's an worship to the Narayan who holds the universe, just as. Now I would like to welcome on the stage our Shayan Patro, Shormishta Dashgupta, Disha Bishas, Krishna Shahu, Mouma Maiti, Dishani Mukherjee, Indrani Bhattacharya, Shayani Acharya, Shangita Patro, Akshay Moody, Tanmay Roy, Titli Panchali, Riya Kaur, Ananya Datto and Tufan Shayali.
On this very special evening, let us tell you a story of beyond our creation, even beyond the creation of our universe. A trillion of years ago, there was just nothing but a woman who kept carefully a golden egg inside her womb. According to Hindu mythology, Brahma was known as Hiranyagarbha or the golden egg. Brahma, the creator of this entire universe, was slept within this egg for thousands of ages, with all of his creation held within the egg. Then he awake and came out to meet gods, daityas or demons, asuras or titans, universe, planets, lands, oceans, islands, everything, just from an egg. So even God of the gods himself was born from an egg. The entire universe was created from a woman's egg. And the one who carried that egg inside her womb, we call her Mohamaya or Adi Shakti, the mother of the universe, presenting According to Brahma Puran, Shatarupa is regarded as the first woman to be created by Brahma to marry Manu, the first man, and their descendants are called Manushva or the human being. Do you know the meaning of Shatarupa? This means Devi Durga. So we all are the part of Devi Durga. Many ages have passed since this incident. আজ আমরা সভ্য সমাজের বাসিন্দা। Now, let's have a look on our mother planet to see what is happening with all our Durgas. Yes, they all are Durgas because they all help to create or develop this planet by different ways, just like the ancient time. আজও দুর্গারা শক্ত হাতে ধরে রেখেছে আমাদের এই ধরণীকে। হাজারো প্রতিকূলতার সত্ত্বেও তারা পিছু পা হয়নি। They have so many boundaries, so many oppositions, but still, they have that courage to be fearless. হয়তো এই সকল প্রতিকূলতা জয়ী নির্ভীক নারীদের জন্যই আমাদের বিশ্বকবি রবীন্দ্রনাথ ঠাকুর লিখেছিলেন,
যদি তোর ডাক শুনে কেউ না আসে তবে একলা চলো রে, যদি তোর ডাক শুনে কেউ না আসে তবে একলা চলো রে, তবে একলা চলো একলা চলো একলা চলো একলা চলো রে, তবে একলা চলো একলা চলো একলা চলো একলা চলো রে। যদি কেউ কথা না কয় ওরে ওরে ও ভাগা, কেউ কথা না কয়, যদি সবাই থাকে মুখ ফিরায় সবাই করে ভয়, যদি সবাই থাকে মুখ ফিরায় সবাই করে ভয়, তবে পরাণ খুলে, তবে পরাণ খুলে তুই মুখ ফুটে তোর মনের কথা একলা বলো রে, ও তুই মুখ ফুটে তোর মনের কথা একলা বলো রে, যদি তোর ডাক শুনে কেউ না আসে তবে একলা চলো রে।
হায় রে মানব সমাজ, or maybe I was wrong. It was not the human society, it is the patriarchal society, who always wants to keep women in chains. Condolences for those who thinks women are the weakest one. It hurts whenever we saw someone thinks women can only used as commodities, অর্থাৎ পণ্য বা ভোগ্যবস্তু, and this is their only contribution to this world. আর এই সব নররূপী ডিমান্ডসদের জন্যই অকালেই ঝরে পড়ে বহু প্রাণ। Oh God, they are not even safe in their own world. In that world, yes, I repeat, that world, which is the most beautiful creation of a woman. আর কবে কিভাবে তারা রুখে দাঁড়াবে এই সম্পূর্ণ পুরুষতান্ত্রিক সমাজের বিরুদ্ধে? Can they ever win or not?
मुझसे मोहन क्यों है रूठे रूठे मोहन यूं कैसे मनाऊं है कैसे मनाऊं क्यों न बोले मुझसे मोहन क्यों है रूठे रूठे मोहन क्यों कैसे मनाऊं है कैसे मनाऊं उन बिन कटे न रहना उन बिन आवे न एक पल चैना उन बिन जियूं तो कैसे मैं जियूं है बहेरा भरे मोरे नैना झरे मोरे नैना मोए नैना सुने नहीं कहना बहे मोरे नैना बहेरा भरे मोरे नैना झरे मोरे नैना मोए नैना सुने नहीं कहना बहे मोरे नैना
When you leave, then you take a little piece of me with you. There's a hole in my soul 'cause I take a little piece of me with you. When you go, when you leave, then you take a little piece of me with you. There's a hole in my soul 'cause I take a little piece of me with you.
যুগে যুগে যতবার সমাজের পাপের ঘরা পূর্ণ হয়েছে ততবারই তিনি এই সৃষ্টিতে অবতীর্ণ হয়েছেন নবরূপে তার সন্তানদের রক্ষা করতে দেবী মহামায়া is the carrier of spirituality, strength, happiness, wisdom. Maybe this is the time to make her awake. Please save us. Save us. Save your child, Ma. We are in grave danger.
आई गिरि नंदिनी नंदित मेधिनी विश्व विनोदिनी नंदनुते गिरिवर विंध्य शिरोधिनी वासिनी विष्णु विलासिनी जिष्णुनुते भगवती हेशिति कंठ कुटुंबिनी भूरि कुटुंबिनी भूरि कृते जय जय हे महिषासुरमर्दिनि रम्यकपर्दिनि शैलसुते करमुरलीरव बीजित पूजित लज्जित कोकिल मंजुमते मिलित पुलिंद मनोहर गुंजित रंजित शैल निकुंजगते जय जय हे महिषासुरमर्दिनि रम्यकपर्दिनि शैलसुते
She is not only the mother of this universe, she is our mother also. Ma cannot refuse her own child's prayer. She is the carrier of strength. She makes us believe that we all made with her strength and energy. So don't wait for others to help you. Just believe in yourself and be your own helper. নিজের সকল জড়তাকে কাটিয়ে উঠে নিজের মধ্যের দুর্গাকে বের করে আনো। তোমার বুকের ভিতর যে আগুন জ্বলছে তাতে পুরে ছাই হোক এই সমস্ত অসুরেরা, বাট দ্যাট ইজ নট সো ইজি। সত্যের পথ, বাঁচার লড়াইয়ের পথ এত সহজ নয়। They all carry demons' blood inside their veins. Can't you see the demons' shadow inside of them? They are the real asuras. যুগে 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 অসুরও নিজের রূপ বদলাচ্ছে আর বসবাস করছে আমাদের এই সভ্য সমাজে, আমাদেরই আশেপাশে। যতবার সমাজে অধর্ম হবে, নারীত্বের অসম্মান হবে, তখনই মা আসবেন। মা আসবেন আমাদের সঠিক পথ দেখাতে। আমাদের মধ্যে থাকা ভয়হীনতা বা ফিয়ারলেসনেসই হলো আমাদের মধ্যের মহামায়া। আর এই মহামায়া নারী শরীরে ততবারই জাগ্রত হন যতবার নারীর নারীত্বে আঘাত লাগে।
श्रीनन्तु विश्वे अमृतस्य पुत्र आयेधानि दिव्यानि वस्तु वेदाहमेतं पुरुषं महन्तम् आदित्यवर्णं तमसा परस्तात् श्रीनन्तु विश्वे अमृतस्य पुत्र आयेधानि दिव्यानि वस्तु वेदाहमेतं पुरुषं महन्तम् आदित्यवर्णं तमसा परस्तात्।
It was an awesome performance by the team, truly holding up a meaning to the Sanskrit slok of Sarva Mangalya Mangalye Shive Sharvat Shadite Sharanye Trambake Gauri Narayani Namastute. Thank you to those who performed this act. Really, you deserve an appreciation.
Hello to all, and once again a warm welcome to all the events. I am Anjan Khamroy,
here with my partner Deep Chattaraj for holding up their further continual of events organized.
Songs are melodies that either or expresses the motive of thoughts and vibes. Let us submerge into the coming up song Amar Bhitoro Bahire, composed by Rudra Muhammad Sahidullah and to be performed on stage by Shriti Ghosh, Suparna Das, Barsha Bera. We do welcome you over stage to show your performance.
Hello, hello.
ঢেকে রাখে যেমন কুসুম পাপড়ির ডাব ডালে ফসলের ঘুম ঢেকে রাখে যেমন কুসুম পাপড়ির ডাব ডালে ফসলের ঘুম তেমনি তোমার নিবিড় চলা তেমনি তোমার নিবিড় চলা মরমের মূল পথ ধরে আমার ভিতর বাহিরে অন্তরে অন্তরে আছো তুমি হৃদয় জুড়ে আমার ভিতর বাহিরে অন্তরে অন্তরে আছো তুমি হৃদয় জুড়ে है ये तारीफें दिल से जो मैंने करी है जो तू मिला तो सजी है दुनिया मेरी हमदम ओ आसमां मिला ज़मीं को मेरी आधे आधे पूरे हैं हम तेरे नाम पे मेरी जिंदगी लिख दी मेरे हमदम আমার ভেতর বাহিরে
Thank you so much, guys, for performing such a soothing performance on the stage. Coming up, we have a dance performance. We define dance as an expression flowered up physically to express the rhythm of the melody behind.
Now, let's welcome Arunima Chandra on stage to cite up her performance with a mess-up melody behind.
सामने चूल्हा जल रहा है, धुआं आंखों में लग रहा है, जलन भी है, आनंद भी। बस आते ही होंगे, जल्दी से संध्या पूजा हो जाए। जानती है वो क्या कहेंगे।
जैसी हो वैसी ही आ जाओ, सिंगार को रहने दो। जैसी हो वैसी ही आ जाओ, सिंगार को रहने दो।
कहने को जश्न बहारा है, इश्क ये देख के हैरा है। कहने को जश्न बहारा है, इश्क ये देख के हैरा है। फूल से खुशबू खफा खफा है गुलशन में, छुपा है कोई रंज फिजा की चिलमन में। सारे सहम नजारे हैं, सोए सोए वक्त के धारे हैं, और दिल में कोई कोई सी बातें हैं। ओ कहने को जश्न बहारा है, इश्क ये देख के हैरा है। फूल से खुशबू खफा खफा है गुलशन में, छुपा है कोई रंज फिजा की चिलमन में।
ये नरम नरम नशा है, बढ़ता जाए। कोई प्यार से घुंघटिया देता उठाए। अब बावरा हुआ मन, जग हो गया है रोशन। ये नई नई सुहागन हो गई है तेरी जोगन। कोई प्रेम की पुजारन मंदिर सजाए। सैया অল্প দুটো আছে তোমার, ঠিক আছে?
After citing such a performance mixed with both expression and energy, we have another group to be brought on stage with vibes and hypes. We would love to welcome the performing participants, Joyashri Beda, Ashrita Samanta, Shriti Ghosh and Sonia Patro on stage to perform on a dance of different mashups from English industries.
ABC 도레미 만큼 착했던 나 그 눈빛이 싹 변했지 어쩜 이 또 하나니까 간파른 나비처럼 날아가 잡지 못한 건 넌 네 몫이니까 활짝 꽃피웠던 시간도 이제 모두 내게 lie 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 붉게 다 버려진 너와 난 괜찮아 넌 괜찮을까 구름 한 점 없이 예쁜 날 꽃향기만 남기고 갔단다 꽃향기만 남기고 갔단다 I love it when you call me señorita. I wish I could pretend I didn't need ya, but every touch is ooh la la la. It's true la la la. Oh, I should be running. Oh, you keep me coming for ya.
Land in Miami, the air was hot from summer rain, sweat dripping off me. Before I even knew her name, la la la. You felt like ooh la la la, yeah, no.
Come on, turn the radio on. It's Friday night and I won't be long. Gotta do my hair, put my makeup on. It's Friday night and I won't be long till I hit the dance floor, hit the dance floor. I got all I need. No, I ain't got cash, but I got you, baby. Baby, I don't need dollar bills to have fun tonight. Baby, I don't need dollar bills to have fun tonight. I don't need no money as long as I can feel the beat. I don't need no money as long as I keep dancing. Tómame la mano que tú eres cosa buena. Esta noche quiero bailar sobre la arena. No sé una ajena, no sé nada más. Ven pa' acá, y ahora tómame la mano que tú eres cosa buena. Esta noche quiero bailar sobre la arena. No sé una ajena, no sé nada más. Ven pa' acá, y ahora give me boy, I'm your lady. I can hear what you want, feel so crazy. Give me boy, I'm your baby, I'm your lady. I'm your girl, I'm your mama. Come on boy, don't be shy, don't want drama. I'm your girl, Bella, sexy mama. You start this fire, the way you look at me takes me higher. I want this moment to last forever and ever and over again. Give me some of that, ooh la la. Tómame la mano que tú eres cosa buena. Esta noche quiero bailar sobre la arena. No sé una ajena, no sé nada más. Ven pa' acá, y ahora tómame la mano que tú eres cosa buena. Esta noche quiero bailar sobre la arena. No sé una ajena, no sé nada más. Ven pa' acá, y ahora.
Seeking the reactions from audience, all I could understand that it was a great build up to the energy over the stage by you all guys. Thank you so much. Now, we are yet to have another dance performance.
So let's continue further enjoying by welcome Preeti Dinda and Suparna Das on stage to show up their performance on another energetic mess-up on Hindi songs.
एक तू है, एक जान है, दोनों तुझ पे, दोनों तुझ पे कुर्बान है। एक मैं हूँ, एक ईमान है, दोनों तुझ पे, हाँ तुझ पे, दोनों तुझ पे कुर्बान है। मेरा प्यार भी तू, मेरी बात साथ, जज्बात भी तू, परवाज़ भी तू, एहसास भी तू, मेरी सांस नाच और हयात भी तू, मेरा राज भी तू, मेरी बात प्यास और लिबास भी तू, मेरी जीत भी तू, मेरी बात भी तू, मेरा राज नाच और लिबास भी तू।
सुनते हैं जब प्यार हो तो दिए जल उठते हैं, तन में मन में और नयन में दिए जल उठते हैं। आजा पिया आजा, आजा पिया आजा हो, आजा पिया आजा, तेरे ही लिए जलते दिए, बितानी तेरे साए में, साए में जिंदगानी बितानी तेरे साए में, साए में। नारे 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 नन्ना नारे 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 नन्नारे 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 नन्नारे। बरसो रे मेघा मेघा, बरसो रे मेघा मेघा, बरसो रे मेघा बरसो, बरसो रे मेघा मेघा, बरसो रे मेघा मेघा, बरसो रे मेघा बरसो। मीठा है कोसा है, बारिश का बोसा है, कोसा है कोसा है, बारिश का बोसा है। मीठा है कोसा है, बारिश का बोसा है, कोसा है कोसा है, बारिश का बोसा है। जल 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 थल जल थल जल जल जल
awesome maintenance of the flow of energy. I could see a lots of movement behind. So now let's cite up a fixed fusion song to be performed by Shayoni Acharya on stage. We do welcome you, Shayoni, on stage to show your performance. Shayoni, once again I call you up. Okay, fine. Skipping up, we do call up the performance to balance our energy after watching some whelming dances. Let's call Shriti Ghosh and her group to perform on the song Amay Proshno Kore Nil Dhrubo Tara by Salil Chaudhary.
Hello. Hello, hello. कहीं दूर जब दिन ढल जाए साँझ की दुल्हन बदन चुराए चुपके से आए कहीं दूर जब दिन ढल जाए साँझ की दुल्हन बदन चुराए चुपके से आए मेरे ख्यालों के आंगन में कोई सपनों के दीप जलाए कहीं दूर जब दिन ढल जाए साँझ की दुल्हन बदन चुराए चुपके से आए কারা যেন ভালোবেসে আলো জ্বেলেছিল সূর্যের আলো তাই নিভে গিয়েছিল কারা যেন ভালোবেসে আলো জ্বেলেছিল সূর্যের আলো তাই নিভে গিয়েছিল নিজের ছায়ার পিছে ঘুরে ঘুরে মরি মিছে একদিন চেয়ে দেখি আমি তুমি হারা कहीं दूर जब दिन ढल जाए साँझ की दुल्हन बदन चुराए चुपके से आए
জবাব কিছুই তার দিতে পারি নাই, শুধু পথ খুঁজে কেটে গেল এ জীবন সারা, এ জীবন সারা। আমায় প্রশ্ন করে নীল ধ্রুব তারা, আর কত কাল আমি রব দিশাহারা, রব দিশাহারা। থ্যাংক ইউ।
Thank you guys for showing up this performance.
To again bring the expression with melody, we welcome Debosmita Senapati on stage to perform her dance on Jia Jale song from Bollywood platform.
जिया जले जा जले, जिया जले जा जले, नैनु तले धुआं चले, धुआं चले.
പുഞ്ചിരി തഞ്ചിക്കൊഞ്ചിക്കോ മുന്തിരി മുത്തൊളി ചിന്തിക്കോ മഞ്ചണി വർണ്ണ ചുന്ദരി വാവേ തങ്കുന്നക്ക തകതിമിയാടും തങ്കനിലാവേ ഹോയ്. പുഞ്ചിരി തഞ്ചിക്കൊഞ്ചിക്കോ മുന്തിരി മുത്തൊളി ചിന്തിക്കോ മഞ്ചണി വർണ്ണ ചുന്ദരി വാവേ തങ്കുന്നക്ക തകതിമിയാടും തങ്കനിലാവേ.
जिया जले जा जले, नैनु तले धुआं चले, धुआं चले. रात भर धुआं चले, जानू ना जानू ना जानू ना सके रे. जिया जले जा जले, नैनो तले धुआं चले, धुआं चले. रात भर धुआं चले, जानू ना जानू ना जानू ना सके रे. जिया जले जा जले. देखते हैं तन मेरा, मन में चुभती है नजर. देखते हैं तन मेरा, मन में चुभती है नजर. होठ सिल जाते उनके नर्म होठों से, मगर गिनती रहती हूँ मैं अपनी करवटों के सिलसिले. क्या करूँ कैसे कहूँ, रात कब कैसे ढले. जिया जले जा जले, नैनु तले धुआं चले, धुआं चले. रात भर धुआं चले, जानू ना जानू ना जानू ना सके रे. जिया जले जा जले, नैनु तले धुआं चले, धुआं चले. रात भर धुआं चले, जानू ना जानू ना जानू ना सके रे. जिया जले जा जले.
Thank you, Debosmita, for maintaining a wonderful energy.
Now I would like to welcome Shayoni Acharya once again on stage to perform her fixity fusion to maintain the another flow of energy.
ফুল নতুন কানের দুল বাঁধলো চোখে চুল বিবিয়ানা ये सारी कोयलें बनी है आज डाकिया कुहू कुहू में चिट्ठियां पढ़े मजाकिया मगा सनी धा 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 नी सनी रे गा मा धा पा सनी सा धा नी पा सारी कोयलें बनी है आज डाकिया कुहू कुहू में चिट्ठियां पढ़े मजाकिया इन्हें कहो कि ना छुपाए किसने है लिखा बताए उसकी आज मैं नजर उतार लूं हे हो पियाली रे हे हो दुलाली रे हे हो पियाली रे हे हो दुलाली रे নতুন বিয়ের ফুল ফুটেছে খালি পাখালি মনে একটা কোণে নতুন বিয়ের ফুল ফুটেছে ইনি বিনিটা পাহাতিনিটা নাটুনিটা সা সাহেব বাবুর বউছে দেখতে ভারী খাসা উলো কুটি খুলো কুটি ঘোরি है नोची তোর বাবালে বুড়া মোর করব আমি কি সবার লু সবার লু সবার লু সবার লু সবার লু হাই সবার লু নতুন বিয়ের ফুল নতুন কানের দুল বাঁধলো চোখে চুল বিবিয়ানা
Thanks a lot, Shayoni Acharya, with tons of expression and movements. You really impressed the audience here. Now, I would like to welcome Moma Maiti on stage to perform to maintain the flow of energy, further with a dance on the melody Bahu Manarath by Shubhamita Banerjee.
बहु मनोरथ साजू अभिसारे पहलू सुनील बेस बहु मनोरथ साजू अभिसारे पहलू सुनील बेस काजर नयने सलाज बयाने कुसुमे सजानु केस सखी हम मोहन अभिसारे जाऊं बोलो हम एतक सुख कहाँ पाऊं जमुनार पार गहन आधार घन नहीं पवन माझे पिया से था मोर वेदना कातर मोहे लागे बैठे आछे सखी हम मोहन अभिसारे जाऊं बोलो हम एतक सुख कहाँ पाऊं सखी चिरा भागिनी हम बैठे काकिनी पोहानु रजनी तबुनाइलो श्याम सखी चिरा भागिनी हम कृष्ण कजल पिघल सजल नयन निरधारे कठिन पथ वृथ मनोरथ विफल अभिसारे सखी हम कबहु न अभिसारे जान दुखला एतत सहन नहीं पाऊं वृथा मनोरथ साजो अभिसारे पहलु सुनिल वेश काजर नयान सलाज बयानी कुसुमे सजानु के फिर आजू मोहन अभिसारे जान सखी बोल एतक दुख कहाँ पाऊं
By showcasing the emotion as well as the dance, we would like to provide you a thanks, Moma Maiti.
Let's welcome to stage Krishna Sau to showcase another performance of dance on song Ghar Mare Pradesiya by Bisali Mahade.
रघुकुल रीत सदा चली आई रघुकुल रीत सदा चली आई प्राण जाए पर वचन न जाए
It was an awesome display of expression and movements being channelized together. Now, being pleased with the performances on the stage, I would like to welcome Swagata Mishra to perform on the song Mere Dolna by Shreya Ghoshal and M.G. Sri Kumar. Please do maintain silence. This is a seminar hall.
सुन मेरे प्यार की धुन मेरे ढोल ना सुन मेरे ढोल ना सुन मेरे प्यार की धुन मेरे ढोल ना सुन मेरी चाहते तो फिजा में बहेंगी जिंदा रहेंगी होके फना मेरे ढोल ना सुन मेरे प्यार की धुन मेरे ढोल ना सांसों में तेरे सरगमे हैं अब रात दिन जिंदगी मेरी तो कुछ ना अब तेरे बिन
सांसों में तेरी सरगमे हैं अब रात दिन जिंदगी मेरी तो कुछ ना अब तेरे बिन
तेरी धड़कनों की सरगोशी मेरी धड़कनों में बजती है
मेरी जागती निगाहों में ख्वाहिश तेरी ही सजती है
मेरे ख्याल में हर पल तेरे ख्याल शामिल है
लम्हे जुदाइयों वाले मुश्किल बड़े ही मुश्किल हैं निजा 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 धनी 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 पदा 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 गम पदा नि निजा pani niza pani niza pani niza pani niza gama pada nida pada nida pada niza
gama pada nida pada nida pada niza
gama pada nida pada pama gama gari sani sani sa gama ga sa gama pada pada niza
gama pada nida pada pama gama gari sani sani sa gama ga sa gama pada pada niza
niza niza niza niza niza
thani 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 thani
gama pada nida pama gama pada nida pama
gama pama gama gari sare sani dhani sa
pada ni ni sa sa ga gama ga
sa sa ga gama pada dhani da
dhani sa ga dhani 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 da pama gama dhani sa dhani da pama gama gari sani dhani sa gama dhani sani da pama gama gari sani da gama dhani sani da pama gama gari sani da dhani ni sa sa ga ni ni sa sa ga mama sa sa ga mama da da ga mama da da ni ni mama da da ni ni sa sa da ni ni sa sa ga
मेरे ढोल ना सुन मेरे प्यार की धुन मेरे ढोल ना
No words to explain this performance that we cited recently. It was a flow of energy maintained with an everlasting smile on the stage. Now, being an Indian, especially being a Bengali, folk performance are an emotions to us, right? So, let's satisfy our emotions.
Let's welcome the group of Miss Titli Panchali, Disari Mukherjee, Indrani Bhattacharya, Sayoni Acharya, Songita Patro, Onnesa Khatun, Tabsum Khatun, Onnesa Dotto and Riya Kaur on stage to perform the emotion of folk performance.
हर खाए हय्यू हाय हाय आजानी किरणों बिखराए उमंग भी छलका है मन हर रंगी परोड़ आवी खुशियों संगे लावी हर खाए हय्यू हाय हाय आजानी किरणों बिखराए उमंग भी छलका है मन हड़वे थी गुनगुनाए हाय 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 हाय
हे शुभारंभ हो शुभारंभ मंगल बेला आई सपनों की डहरी पे दिल की बाजी रे शहनाई 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 ख्वाबों के बीज कच्ची जमीन पे हमको बोना है आशा के मोती सांसों की माला हमें पिरोना है अपना बोझ मिल के साथ ही हमको ढोना है शहनाई शहनाई शहनाई
रास रचिलो साज सजिलो शुभ घड़ी छे आवी आछा आछा टमटमाता शमणाओ छे लावी ओ लावी ओ लावी रंगी परोड़ आवी खुशियों संगे लावी हर खाए हय्यू हाय हाय रंगी परोड़ आवी खुशियों संगे लावी हर खाए हय्यू हाय 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 हाय
हाँ मजा है जिंदगी नशा है जिंदगी धीरे धीरे चढ़ेगी हाँ दुआ दे जिंदगी बता दे जिंदगी बात अपनी बनेगी हाँ ख्वाबों के बीज कच्ची जमीन पे हमको बोना है आशा के मोती सांसों की माला हमें पिरोना है अपना बोझ मिल के साथ ही हमको ढोना है शहनाई शहनाई शहनाई
धिन तड़ाक आजा और के सराट पैरों से बीड़ी जरा खोल नगाड़े संग ढोल बाजे धाए धाए धम धम धाए नगाड़े संग ढोल बाजे धाए 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 धम धाए ये धिन तड़ाक आजा और के सराट पैरों से बीड़ी जरा खोल नगाड़े संग ढोल बाजे धाए धाए धम धम धाए नगाड़े संग ढोल बाजे धाए 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 धम
ওই খুনি মন পিছলি ওই যায় ওই গাঁৱৰ ডেকা গাভৰু ওই আহিল সাজি কাছি ওই গুৰু গোসাঁই কাৰো গাত নাই ওই এ বোলে বিহু চেক চেকি ধিনকিতি ধিন দাও দাও ও সে বোলে বিহু চেক চেকি ধিনকিতি ধিন দাও দাও ধিনকিতি ঢোলে বাও ওই তি তলপে পাবা ওই ধিনকিতি কালো জলে কুচলা তলে ডুবলো সনাতন আইজ সারা না কাল সারা না পাই যে দর্শন কালো জলে কুচলা তলে ডুবলো সনাতন আইজ সারা না কাল সারা না পাই যে দর্শন নদী ধারে চাষে বধু মিছাই করোয়াস ঝিরি হিরি বা কালো দি বইছে 12 মাস নদী ধারে চাষে বধু মিছাই করোয়াস ঝিরি হিরি বা কালো দি বইছে 12 মাস কালো জলে কুচলা তলে ডুবলো সনাতন আইজ সারা না কাল সারা না পাই যে দর্শন মেদিনীপুরে রায়না ছিরন বাঁকুড়া রই ফিতা যতন করে বাদলি মাথা তাও যে বাঁকা সিথা মেদিনীপুরে রায়না ছিরন বাঁকুড়া রই ফিতা আরে যতন করে বাদলি মাথা তাও যে বাঁকা সিথা পেছ পাড়িয়া রাজকুমারী গলায় চন্দ্রহার দিনে দিনে বাড়ছে তোমার চুলেরই বাহার পেছ পাড়িয়া রাজকুমারী গলায় চন্দ্রহার দিনে দিনে বাড়ছে তোমার চুলেরই বাহার কলি কলি ফুল ফুটেছে নীল কালো আর সাদা কোন ফুলেতে কেষ্ট আছে না কোন ফুলেতে রাধা কলি কলি ফুল ফুটেছে নীল কালো আর সাদা কোন ফুলেতে কেষ্ট আছে না কোন ফুলেতে রাধা কালো জলে কুচলা তলে ডুবলো সনাতন আজ সারা না কাল সারা না পাই যে দর্শন নদী ধারে চাষে বধু মিছাই করোয়াস ঝিরি হিরি বাঁকা নদী বইছে 12 মাস কালো জলে কুচলা তলে ডুবলো সনাতন আজ সারা না কাল সারা না পাই যে দর্শন লালা লালাই পলাশ বন মন হইল বাজু চাটন লালা লালাই পলাশ বন মন হইল বাজু চাটন মন মেতেছে মহুল ফুলের বাসে গো মন মেতেছে মহুল ফুলের বাসে আমার মন মেতেছে মহুল ফুলের বাসে গো মন মেতেছে মহুল ফুলের বাসে ওমর সজনী গোপনে সে আমায় ভালোবাসে গো গোপনে সে আমায় ভালোবাসে ওমর সজনী গোপনে সে আমায় ভালোবাসে গো গোপনে সে আমায় ভালোবাসে তার ভালোবাসার নাই ঠিকানা যখন তখন না গোনা ভালোবাসার নাই ঠিকানা যখন তখন না গোনা চিতদিনের জানা শোনা থাকে আশেপাশে গো থাকে আশেপাশে মন মেতেছে মহুল ফুলের বাসে গো মন মেতেছে মহুল ফুলের বাসে ও সজনী গোপনে সে আমায় ভালোবাসে গো গোপনে সে আমায় ভালোবাসে ওমর সজনী গোপনে
গোপনে সে আমায় ভালোবাসে গো গোপনে সে আমায় ভালোবাসে লালে লাল হয়ে পলাশ বন মন হলো বাজু চাটন লালে লাল হয়ে পলাশ বন মন হলো বাজু চাটন মন মেতেছে মহুল ফুলের বাসে গো মন মেতেছে
আরে মিত্র ভানু গন্তিয়ার কথায় প্রভু দত্ত প্রধান করিল সুর এত সুন্দর গীত আমাদের এই গীত একটু তোমাদের ভাষায় শোনাবো নাকি গো রঙ্গবতী রে রঙ্গবতী রঙ্গবতী রে রঙ্গবতী 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 রঙ্গিলা অন্তরে হাবুডুবু তোমারি কথা রঙ্গবতী রঙ্গবতী রঙ্গিলা অন্তরে হাবুডুবু তোমারি কথা
হায় গো লাজে 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 হায় গো লাজে 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 হায় লাজে লাজে মোরে দোহাই রসিক নাগর মনে নাই কোনো ব্যথা লাজে লাজে মোরে দোহাই রসিক নাগর মনে নাই কোনো ব্যথা
স্বপ্ন দিলে স্বপ্ন
স্বপ্ন আবার দিলে স্বপ্ন
রূপে তোমার মরি বাছি বক্ষ জুড়ে হাহাকার রঙ্গবতী রঙ্গবতী ও রঙ্গবতী রে রঙ্গবতী ও রঙ্গবতী রে রঙ্গবতী প্রিয়া প্রিয়ার নলক হাতে কাকন মন বিহঙ্গন আমার জীবন ধুলায় উড়ি যায় রে প্রিয়া প্রিয়ার নলক হাতে কাকন মন বিহঙ্গন আমার জীবন ধুলায় উড়ি 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 যায় রে
মুখে কোনো কথা নাই যে গো মুখে কোনো কথা নাই যে গো মুখে কোনো কথা নাই শুধু আকারে সাকারে আখির বাহারে সরষার খেতে দুইজন গো মন আকারে সাকারে আখির বাহারে সরষার খেতে ভাসে গো
ডুবে 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 আমার অন্তর পুড়িল ডুবে 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 আমার অন্তর পুড়িল
কুহু কুহু ডাকে আবার বুকের মাঝে হয়তো
রঙ্গবতী রঙ্গবতী ও রঙ্গবতী রে রঙ্গবতী ও রঙ্গবতী রে রঙ্গবতী হায় রঙ্গবতী রঙ্গবতী রঙ্গিলা অন্তরে হাবুডুবু তোমারি কথা রঙ্গবতী রঙ্গবতী রঙ্গিলা অন্তরে হাবুডুবু তোমারি কথা
Now we are at our end of the programs held. All that adrenaline rush will be going down now. So I would like to thank all the performers as well as the reverend guests and teachers, and to our dear students.